Assalamualaikum warahmatullahi wabarakatuh. Dengar?
Dengar.
Dengar Doktor.
Okey, good.
Dengar dah.
So siapa yang present first? Siapa present
Doctor,
first?
I'm the first presenter.
Okey, okeylah kita mula dengan membaca Al-Fatihah. Okeylah, boleh start kot?
Okay. Assalamualaikum. Before that, all of you can see my slide.
Yes.
Okay. Assalamualaikum and good morning. I bid to our supervisor, Dr. Alwi, and my friends. Today, our group will present about genetic, and for my part, I will present on the molecular basis of genetic. As for my part, BMS knowledge. Okay, before we begin, I just a brief on my outline. We go first to the introduction, chromosome, genes and allele, DNA, mutation, and importance of molecular genetic. So as the introduction, genetics means study of heredity, which is controlled by a vast number of factors called genes. And for the molecular biology, is study of cell processes and codes in living organism. Meanwhile, for the molecular genetics, is the field of biology that studies the structure and the function of gene at a molecular level. For this seminar, I will more concern on the molecular genetic. So as you can see in the slide, in the cells, there is a nucleus, and in the nucleus consists of chromosome. As we know, chromosome made of DNA, and a segment of DNA is called gene. So from this chromosome, and we'll make the DNA, and it will synthesize a protein. So as a begin, we need to know the chromosome. Okay, what is chromosome? Chromosome is a fine thread-like structure of nucleic acid, DNA, and protein found in the nucleus of cells. In other words, is histone. So, in simple word, chromosome is made up of DNA and histone. So, chromosome carries a genetic information in form of gene. So, one chromosome consists of two identical chromatids. In the picture, you can see the chromatid consists of P arm and Q arm. P arm is for the short arm and Q arm is for the long one. And the constricted point is the centromere. Okay. Okay. As we all know, every human cell has 23 pairs of chromosome, total of 46 chromosome, and 22 pairs is autosome, and one pair is sex chromosome. XX is for the female and XY is for the male. And we copy of each chromosome is inherited from each parent. Okay, in the picture you see the 22 pairs of autosome and sex chromosome. This is the normal human karyotype. Okay, as for the gene as mentioned before, gene is a segment of DNA that code for a particular protein. In other words, is trait. So you can see the picture in the lower side. In the lower side consists of gene that that coding the trait of brown eyes. Okay, and for the allele is a gene found in the same locus on the homologous chromosome, which control the same characteristic. You can see the picture in the left side as for the allele for the blue eyes is the blue one, and the brown is for the brown eyes. And in the individual B, it has a homozygous allele for the brown eyes, and for the individual C, it has a homozygous recessive, as he only has a blue colored eyes, a blue colored allele. Okay, okay. For the DNA, DNA, as name implies, is the oxyribonucleic acid. Is a polymer of nucleotide covalently linked by 3' to 5' phosphodiesterase bond. So DNA is a double-stranded structure, double helix, as you can see on the figure. And uh, as the name implies, the oxyribonucleic acid is made out of nucleic acid, consists of three basic components: the oxyribose sugar, phosphate group, and nitrogenous base. Nitrogenous base consists of four: adenine, guanine, cytosine, and thymine. Okay, all of you know the function of DNA, so I just revise. DNA is responsible in terms of long-term storing and retrieve the information, and it's basically perform two main vital process, which are replication and transcription. Replication is a genetic information processed by a parents transferred to the progeny or daughter cell or offspring. In other words, replication, we want to do the same exactly the gene. And for the transcription, the source of information used for the synthesis of all protein molecule of the cell and the organism. Okay, in simple understanding, you can see the picture from DNA. We do the replication to do more DNA, and we need to undergo transcription to do the RNA and do the translation to make a protein. Okay, okay, for the replication of DNA is a revision from our previous BMS knowledge. Okay, I hope you guys can focus. Okay, for DNA replication, the hero one is the helicase enzyme that break the bond, unwind the parenteral double helix, and when there is a break the bond and there is presence of single strand binding protein. Single strand binding protein SSBP will stabilize the unwound parenteral DNA. So the leading strand is synthesized continuously, means at the up at the picture above at the blue color, light blue, it is a leading strand synthesized continuously in the 5' to 3' way direction by the DNA polymerase. So for the other strand, in the lagging strand at the below, lagging strand is the below the blue, light blue, is synthesized discontinuously. The difference is the leading strand is continuously, but the lagging strand is discontinuously, and the primase, we synthesize a short RNA primer, the pink color, which is extended by the DNA polymerase. So DNA polymerase will extend this RNA primer and the lagging strand. So DNA polymerase will form the Okazaki fragment. So there will be several Okazaki fragments. So we want to connect this Okazaki fragment at the lagging strand. We use a DNA polymerase. So you can see the picture. Okay. Next, we do the RNA. As we done the RNA replication, we do the RNA transcription. Means from the DNA, we want to do the RNA. Okay, in the figure, you just focus on the red color of RNA. Okay, the blue one is the DNA. So the first is, we want to the RNA polymerase will unwind the DNA. So you can see the binding of RNA polymerase and local DNA unwinding. So it will initiate the RNA synthesis. As you can see, the RNA for direction is 5' to 3', the pink color at the picture left side, and it undergo the process of elongation. So elongation means RNA become longer, and then further elongation until there is a termination signal. So therefore, it will produce RNA transcript. Okay, as we done the RNA transcription, okay, we want to do the RNA splicing. As we know, RNA is many mRNA, rRNA, tRNA. So for this slide, I will just more focus on mRNA. So RNA splicing is a process that we remove intron and we join the exon together as a primary transcript. So it's catalyzed by the spliceosome. Okay, in the picture is explained more briefly. Okay, remember intron we throw into the dustbin. Exon we want the exon to connect each other. Okay, so we have the matched mRNA. So this mRNA is important to do the uh, protein. So mRNA translation, where the process is from amino acid from mRNA, we coded to form specific amino acid or we known as polypeptide. So codon take or RNA nucleotide that correspond with the specific amino acid or stop signal during the protein synthesis. Okay. Mm, this is just a brief on the how mRNA do the protein. Okay, from the initiation, elongation, and termination. Okay. Next, in this slide, I will more focus on the mutations, the highlighted one, mutation. As mutation is a change occur in DNA sequence. Uh, DNA sequence either due to the mistake when the DNA is copied or as a result of environmental factor. Okay, mutation is a permanent alteration of DNA. So as we go for further in the causes, there is two causes: error made during the meiosis or mitosis, in known as we'll go further on the replication error. For the environmental factors such as chemicals, radiation, UV light will cause the DNA to break down. That will worsen and make the cell produce slightly different DNA. Okay, okay. There is classification of mutation divided into three: genome, chromosome, and gene. As the name implies, uh, genome is the whole our chromosome. So it's a permanent change in the genetic makeup of organism. Okay, the second part is chromosome, means chromosome itself is affect. So affect the whole gene on the chromosome. So it's further divided into four main types: duplication, deletion, inversion, and translocation. So you can see the picture. For the chromosome mutation, okay, there is deletion. Okay, as deletion, there is a part of chromosome is left out and the deletion is occur. As you can see, the D is delete, so we will have ABCE. Okay, when for the insertion, when the chromatid is break off and attached to the sister chromatid, and insertion is occur where the gene B and C. So there is extra A B C B A B A B C B C D E. So extra chromosome, extra gene in the chromosome. Okay, when for the inversion, inversion is happen when a part of chromosome is break off and reattach back, mean backward, and inversion is occur. Instead of A B C D E, will happen A D C B E. So for the translocation is a famous one. Translocation when a part of one chromosome is break off and added to the different chromosome. You can see the part of the WXY. Instead, is translocate in the long the another chromatid. It will happen WXABCDE. Okay, this translocation happen. Okay, next we go for the gene mutation. Gene mutation means the mutate is happen in the gene. So there is three: point mutation, frame shift mutation, and trinucleotide repeat mutation. Okay, as the point mutation, okay, point mutation is have occurred in the single gene. Okay, you can see there is further divided into silent mutation, missense mutation and nonsense mutation. As the silent mutation, codon containing the change base may codon for the same amino acid means there is no difference. For example, for the lysine, TTC, the original one is TTC will happen RNA AAG, so it happen AAA. So it's still a lysine amino acid. For the missense, okay, missense codon containing the change base may code for the another amino acid. It's more worsen. For we need as the normal one is arginine amino acid, but happen another one. Okay, for the nonsense is become change may become a termination one. Uh, it happened to be a stop codon. So it's also worsening the gene. And for the another mutation is frame shift mutation. Okay, frame shift mutation. Okay, you can see the picture. Okay, the above is the normal one, and the below is the mutated one lah. Okay, okay. From the frame shift, it means that one or two nucleotide is either deleted or added into the coding region. So, for example, in cystic fibrosis, the deletion of three nucleotide CTT, phenylalanine, is lost at the position of 508. So there is no phenylalanine amino acid in the chromosome. Okay. Okay. The last one is the trinucleotide repeat expansion. Okay. I just give trinucleotide means try three. So there is a repeated trinucleotide added on the chromosome. So for example is Huntington disease, there is CAG codon. You can see the left side of the figure is for the Huntington disease, and for the fragile X syndrome at the right figure. Okay, fragile X syndrome means extra CGG codon. So at the FMR1 gene, it happened at the X chromosome. That's why the name is fragile X syndrome. Okay, another one is myotonic dystrophy, but I do not show the figure for the myotonic dystrophy. Okay, why we need to know the understand the molecular genetic is basically it gives insight into the heredity, genetic variation and mutation. So it's important for diagnosis, management of the genetic disorder, and we need to identification of disease carrier for the genetic counseling in our clinical practice. Okay, I think that's all from me. Any question?
No, no question.
Okay. Okay, that's a very good presentation. Any question from anyone? This is very important because this is the foundation of genetic and foundation of genetic. The basic of physiology of the genetic. Okay, so basically in general principle, you've got the 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 DNA. Okay, DNA is similar. DNA is a complex polymer, lah. Just similar like complex carbohydrate. Is similar to the protein and also the lipids. So it makes of monomer, kan? The monomer is the nucleotides, kan? Okay, nucleotide is consists of the pentose sugar, the backbone, the phosphate backbone, and also uh, the the what you call it, I can't remember the name. What's the name again?
Nitrogenous base.
Yeah,
Nitrogenous species.
yes. Which divide into two, kan? You ada pyrimidine and purine, kan? Kan? Which
Yeah.
one is pyrimidine? Which one is purine? Because this is very important. Which one is purine, and which one is pyrimidine? You've got four, except thymine and uracil is considered the same. So you've got four. So which one is purine? Which one is pyrimidine? Alia.
Uh, I do not remember, doctor. Sorry. Pu Is it pyrimidine?
No, you got two, purine and also pyrimidine, kan?
Pyrimidine.
Okay, purine is adenine and guanine. Okay, AG is purine. Pyrimidine is the other one lah. TC lah. Cytosine dengan thymine and uracil. So these are purine and pyrimidine. Because why do we have to understand this? Because in some condition, as for example gout kan, you know gout, okay? In gout, what happen is you've got a lot of you
Purine.
yes, you've got a lot of purine kan. You eat a lot of purine containing, because some organism the DNA are made of mainly purines rather than pyrimidine. Okay, purine made of the majority of the nucleotides lah in the DNA. Therefore, they've got a lot of purine when it's break down, so they produce a lot of purine. So things like like beans kan, like nuts, beans, they've got a lot of purine in their DNA. Therefore, if you eat this kind of, patient with gout, they eat food containing a lot of purines, they get gout. Okay kan? Okay, so that's the best thing. And then, okay, what else is important? You have to understand the problem with, the DNA, the mistakes in DNA, especially the process, the replication process, the transcription, and also the translation is occurred every second in our body. So everyone, this occurred every day and every time in everyone, not only in those with cancer or those with autoimmune disease or diseases, but in normal physiology it occurs. But there is a way to prevent this. There is a way to fix this, and there's a way to prevent this. Okay, actually the other important thing, that's why I usually ask the presenter to send me your slides like at least 2-3 days earlier, so that I can see and I can comment, so you can edit a bit. But because last night I'm a bit busy, so I don't have time, and I don't think you've got time to amend. The other important thing is, when you learn about genetic, it's not only the physiology. You have to understand the pathology that can occur on top of the normal physiology, kan? Okay, so it's important, and also it's important part of genetic also. You have to understand the cell cycle, kan? Or do you have any like other lectures on cell cycle? Ada seminar on cell cycle? Because it has to be incorporated together with genetic, basic genetic. Because any mistakes that occur throughout the process of translation, transcription, also replication, okay, this will affect your cell cycle. Okay, kan? So if, for example, any mutation, that is will be detected, any mutation will cause abnormality in the cell lah. Okay, cell that have mutation in their mutation or any abnormality in their DNA, once they're translated, once the protein is translated, they will become defected. Okay, and this will be detected during the cell cycle. Okay, especially during the G phase lah, the G1, G0, G1, G2. Okay, not the M and S phase, because S phase is just preparation. M phase is the process of cell division, but the G phase is actually where they monitor where your body and also the cell itself monitor for any defects. Okay, that's why in G phase there is a gap now. There is gate, what we call it gate. So in cell cycle, you've got two gates, the G0 and G1. I think G1 or G2, sorry, G1 and G2. Some people, some scientists said there's 3, some scientists said there's 4. But what we know now is there's 2 lah, G1 and G2. So during the G1 and G2, okay, this is during the G phase lah. Okay, during the G phase, the cell, okay, the cell will either from, you've got two factors, the external factors and the internal factors within the cell itself. And also the external factors, which is your immune system, will identify any defects in the protein that is produced. That will reflect the cell lah. The cell will become abnormal once the protein abnormal, kan? So if you got a normal immune system and you've got a normal the cell is normal, and you've got the normal, you know, the protogen and also the tumor suppressor gene. Okay, if you've got these two functionings, okay, if you've got these two functionings, then they will actually guard you from abnormality in the cell before the cell division occur. Faham tak? Okay, faham kan? For example, if there's any defect, okay, if there's any defect, so your immune system will determine, oh, this cell is abnormal. So I won't allow this cell to divide. So I will kill this cell. Okay. Or your internal, the internal, like, you know, like the internal, the intracellular mechanism, they also got, each cell they've got their own intracellular mechanism to actually prevent this and to correct this, which is guided by the your oncogene and also the tumor suppressor gene. So if they found out that, I think this, for example, if they found out a cell is abnormal, they will induce apoptosis. Faham kan? Can I mean, you still remember about apoptosis semua kan? So that's why genetic, especially when you're talking about molecular level, like I mean the cellular level protein synthesis, all this you have to relate with cell division, because the main thing is, because in our cell, we have to know the end is actually to produce more cells, to replace the old cell and also to grow. From that, that is the main aim of cell replication. I mean, DNA replication, transcription, translation, all this process. The main aim is actually to produce more protein to allow cell division and to allow growth and also replenish the old, the dead cell, and also for metabolism lah. Faham kan? So okay, so better maybe later on you can read about on cell division and try to link these two together, because you cannot have a basic genetic knowledge without the knowledge of cellular metabolism knowledge, because later on, when you learn about pathology, when you learn about diseases, these two are important, especially in disease like cancer kan, like cancer, in this in immune dysregulation, like autoimmune disease, in genetic disease, any genetic disease, because a lot of genetic disease, they also presented with immune dysregulation kan, and also they also presented with cancer, predisposition to cancer. Patient with some syndrome, they are predisposed to cancer because if the genetic condition affect the proto-oncogene, or affect the tumor suppressor gene, or is affect the your immune system, then definitely you will be prone to cancer. Okay, later, I think later some of you will touch on cancer. Okay, and then it's very important also to understand the concept of point mutation kan. Point mutation, which it can be, you know, when you look at the structure of DNA, your genetic, it start with a nucleotide kan. It start with a nucleotide, single nucleotide, and then it become the trinucleotide, which each trinucleotide will be translated into an amino acid. Come on, satu amino acid is actually translated from a 3 nucleotide. It's not 1 nucleotide become 1 amino acid. It's 3 nucleotide become 1 amino acid. For example, like you've got TTT become lysine, or maybe like CGA become adenine, things like that lah. Okay, TGA maybe become phenylalanine, for example. So you can have defect in single nucleotide that later on will affect the production of your protein, or you can get defect in the 3 nucleotide. For example, like in the expansion kan, CGA, in the trinucleotide expansion, like Huntington kan, Fragile X, like you mentioned lah, myotonic dystrophy kan. So in this condition, the trinucleotide, which usually works together, is expanded. Okay. Or you can also have defect in a larger, you've got larger defect. For example, the whole RNA defect, the whole transcription defect, the whole even the whole lag. The lag and also the continuous defect. Or any of these can be defected, including the chromosome itself. You can get defect in the chromosome, which usually leads to a more prominent presentation lah, syndromic features, rather than usually single gene deletion or like a single nucleotide deletion will lead to more specific diseases. For example, like IEM kan, inborn error of metabolism. Inborn error of metabolism is a genetic disease where most of the inborn error of metabolism is caused by a single gene deletion ataupun single nucleotide defect. Okay. Contoh macam DM kan, diabetes. Okay. There's a lot of diabetes. There's a lot of type of diabetes. One of it is, we call it congenital diabetes. Kan? Congenital diabetes is a single gene defect. It's not even single gene. It's a single nucleotide defect. So defect in one single nucleotide can lead to congenital diabetes, for example, like Duchenne syndrome. Macam tu lah. And then for single gene deletion, single gene defect, one gene deletion or one gene defect can also lead to a disease. For example, like DMD kan, Duchenne muscular dystrophy. This is important because this will actually affect the choice of treatment later. Because I'm sure you heard about gene therapy kan. Okay, gene therapy will not work for chromosome problem. If you got Down syndrome, you cannot do gene therapy because this is whole chromosome. Okay, you cannot use plasmid, or you cannot use a bacteria or virus, lah. Usually, you can use a virus to send in a chromosome because there's no like recessive chromosome. Faham tak? The recessive or the dominant is at the gene level. But for a single gene defect or multiple gene defect, you cannot do that because it's very difficult to, for example, condition like some disease that have multiple gene problems causing contoh macam deletion in one exon, the one whole exon, which consists of few. In one exon, there can be a few genes. So if you lost one exon, you may lose few genes. So this kind of disease will not be curable through genetic therapy, through gene therapy. But disease like Duchenne kan, contoh Duchenne muscular dystrophy is very promising because it's a single gene deletion. So you can actually replace a single gene using virus. Or not. So this kind of disease is more promising and have better prognosis in terms of treatment lah. Okay, faham kan? Apa lagi?
Faham.
Okay, any other questions about the basic genetic? Genetic is very interesting. I know sometimes it's a bit technical, but it's very interesting. And if you understand the genetic, you can understand a lot of diseases lah, a lot of pathology. You can even understand the treatment. Especially if you want to do oncology later, or if you have interest in oncology, you have to be good in genetic. Because if you learn oncology, for example, if you're learning about the chemotherapy kan, the chemotherapy acts differently at different level. Okay, for example like topotecan or apa? Topotecan tu cara sikit. Name me one chemotherapy that you know. Anyone? A common chemotherapy yang you guys come across ke, atau you guys heard about? Anyone? So contoh methotrexate lah. Okay, methotrexate. Okay, how methotrexate works? Okay, how methotrexate works? It's a folic acid Okay, it's a folic acid inhibitor tau. It's a folic acid inhibitor. Okay, the folic acid antagonist. So what it does, because you should understand the structure of your nucleotide kan. Your nucleotide, you've got a phosphate backbone, and then you've got a pentose sugar, and then you've got the purine and pyrimidine kan. So how folic acid works is you need folic acid. Okay, you need folic acid to folic acid. Yes, you need folic acid to build your purine and pyrimidine. You know the folic acid. Tapi itulah to build your purine and pyrimidine. Without folic acid, you can't. So this is how it works. So it stops the cell division by that. And then I'm sure you heard about 6-MP kan, 6-MP make up the purine. I'm sure you heard about 6-TG, thioglobulin, the rest bagi yang common. Saya never lupa semua benda ni. Alkylating agent. Okay, what alkylating agent? How alkylating agent works? So if you understand all this chemotherapy, they works at different stages of cell division and also different stages of nuclear punya ni lah, replication, DNA punya replication and translation. Okay, and you will understand more, and it'll become more interesting lah. It'll become more interesting. Okay lah. I think I said to you. So next presentation. Who's next?
Yes, okay.
Afiq, okay.
Before I start, can you see my slide and can you hear me and see my slide?
Yes.
Yes.
Okay. So my name is Muhammad Syamil Moisuf. And now I will explain about the autosomal disorder. So we will go through this outline, which is first classification, and then Mendelian loss of inheritance, third autosomal dominant disorder, and lastly autosomal recessive disorder. So we know that it is known that the congenital malformation is closely associated with genetic disorder. So geneticists has found out a way to classify the etiology of the genetic disorder, and it can be classified into 5 categories, which is first single gene mutation, which will be later we will discuss later. And then second, chromosomal disorder, which contributes 7% of congenital malformation. And third, multifactorial inherited condition, which constitute 20% of malformation. And then fourth, unusual pattern of inheritance. And lastly, the teratogen exposure, which contribute about 6%. So we will moving on to single gene mutation. So what is single gene disorder? So single gene disorder is a disorder that are caused by mutation in the DNA sequences of a single gene. So it is mainly follow the Mendelian inheritance pattern, which are autosomal dominant, autosomal recessive, and sex linked. So in the Mendelian's law of genetics, it typically describes how characteristic of a trait is passed down to the next generation. So first, the law of dominance. It is characterized by the recessive allele that will be masked by the dominant allele. So and the second one is the law of segregation. The 2 alleles responsible for the trait will be separated and then end up in a different gametes. And lastly, the law of independent assortment, whereby the allele will be distributed independently or by chance during formation of haploid cells. So in autosomal disorder, the gene involved are from 22 pair of the autosome. So it can involve one of the 22 pair of autosome. Unlike the sex linked, it will only involve the sex chromosome. So the single copy of a gene bearing a mutation is sufficient to cause the disease. This is characteristic of the autosomal dominant inheritance. Because according to the first law of the Mendelian inheritance, the dominant allele will manifest the phenotypic manifestation. So it can manifest in either heterozygous or homozygous, and it is transmitted vertically. And the male and female equally affected because it is involved in autosome, not the sex chromosome. And then there is 50% chance of passing mutated gene, as you can see in the figure. So there are several aspects that we have to consider in autosomal dominant disorder. So first, what is penetrance? The penetrance is referred to the degree of obligate carrier of a gene mutation to show clinical sign and symptom. Sometimes, if all patients of autosomal disorder show clinical signs, it means that the disorder, the autosomal dominant disorder, have a complete penetrance. One of the example is neurofibromatosis 1, type 1. And then, however, we have to note that many autosomal dominant disorder show decreased penetrance. So, the second aspect that we have to consider is variable expressivity. Individual carry the same autosomal dominant mutation have different degrees of mutation. It can be from the range of mild to severe degree of mutation. As for from the figure, you can see that the stripe figure, which is the mother, it is actually have the symptom, but typically the mother has mild symptom. So it is this is example of decreased penetrance where the mother have the mother have the disease but show mild phenotypic manifestation, while the son and the daughter show more severe manifestation. And then de novo mutation, it is sometimes appear in a child of uninfected parents, and the spontaneous mutation may be associated with new mutation in one of gametes, and then it is associated with advanced paternal age, which is more than 35 years old. So in de novo mutation, the mutation does not transfer from the mother to the gametes, but the mutation, it is spontaneously initiated during the gametes formation. So it considered as de novo mutation. So these are the example of autosomal dominant disorder. I will explain some of it later in the next slide. So first, we will go through the achondroplasia. So achondroplasia, it is caused by mutation in FGFR3. It is the most common skeletal dysplasia in human, which occur in 1 in 25,000 births. It caused by defect in cartilage derived bone. It will be manifested mainly in bony structure that which will cause short stature, macrocephaly, flat midface with prominent forehead, and the shortening of limbs. So these are the figure of the clinical features of achondroplasia patient. There is macrocephaly, frontal bossing, midface hypoplasia, rhizomelia, trident hand. The one that the trident, the one that the Aquaman possess, and the bowing of the tibia. And then we have to note that there is a there will be a narrowing of foramen magnum. So the patient will have central apnea because of the compression of the brainstem and other increase in intracranial pressure symptoms. So second autosomal dominant disorder that I'll be explaining about is neurofibromatosis type 1. It is caused by mutation in NF1 gene, which code for protein neurofibromin. The penetrance is 100%, which means that all the patients that have the mutated will manifest the symptoms. However, the expression are varied from mild to severe. So it is present in 1 in 3,500 individuals. So to diagnose the NF1, 2 out of 7 criteria must be fulfilled, at least 2, which first, there is presence of 6 or more Kefi outlet spots that is more than 5 millimeters wide. And the second, there is presence of axillary or inguinal freckling, and then leaf nodules in the iris, and then the neurofibromas, and then the optic nerve glioma, and then body defect. And lastly, the family, there is first degree relative that have the same neurofibromatosis type 1. So this is the example of the manifestation of the neurofibromatosis 1. There is bony deformities such as scoliosis and bowing of the tibia. And then there is presence of Kefi outlet macules. And then there is neurofibromas, a cutaneous lesion. It is not associated with the brain. And then there is first degree relative of neurofibromatosis 1. So then we move to the Marfan syndrome. It's caused by mutation in FBN1, FBN1 gene, which code for fibrillin protein. It occurs in approximately 1 in 10,000 individuals. It mainly involves multi-system. But mainly involves 3 systems, which are cardiac, ophthalmology, and skeletal. And many of the symptoms caused by an excess in TGF-beta, the protein usually bound by fibrillin. Especially it affects the cardiac system because if in excess TGF-beta, there will remodeling occur. So these are clinical features of Marfan syndrome, where there is flexible joint, downward slant to eye, and there is thin habitus. There is presence of arenodactyly, and then there is scoliosis. And it also can affect other systems as well, such as cardiovascular, where the aorta dilates, and then there is presence of aneurysm. In eye, it can cause retinal detachment and less dislocation. In lungs, it can cause bulla formation, and the skin, it manifests as a stretch mark. So next, I will explain about autosomal recessive. It manifests only when both copies of the gene affected, parents are not affected, and it is transmitted in horizontal manner, which means that traits can appear in siblings, while in autosomal dominant, the trait appears in parents. And in normal siblings, it can be either carrier or unaffected. And the male and females are equally affected. And it is associated with parental consanguinity, especially the rare disease. And the traits generally involve mutations in genes that code for enzymes. While in autosomal dominant, it essentially involves mutation in regulatory or structural genes. while for autosomal recessive, it involves in mutation in gene that code for enzymes, and is usually associated with serious illness and short lifespan. So in the left figure, it is shown that the figure show autosomal recessive inheritance, where the affected will have where childborn have a chance of 25% that will be affected, 50% will be unaffected carriers, and 25% will be normal. So on the right side, it is shown the pedigree of autosomal recessive inheritance. Typically, the parents will not have the disease, while the child will manifest the disease. So this is an example of autosomal recessive, such as cystic fibrosis, Friedreich ataxia, Gaucher disease, and sickle cell disease. So first, cystic fibrosis. This is one of the autosomal recessive that will be described. So it is caused by mutation in cystic fibrosis transmembrane conduction regulator gene. CFTR is important for proper movement of salt and water across cell membranes and maintaining composition of secretion, mainly in airway, liver, and pancreas. So the abdominal airway secretion make more prone for colonization and reduce the airway defense. As you can see from the airway, figure of airway below, in healthy airway, is in cystic fibrosis airway, there is a lot of mucus secretion because there is impact of sodium and chloride conductance, where there is excess chloride present in the lumen, while sodium is reabsorbed. So there will be the result is there is more thick mucus secretion in the lumen, and typically there will be obstruction in the lumen. So these are the symptoms of cystic fibrosis. And the clinical features include fatigue, chronic cough, recurrent upper respiratory tract infection. There is presence of thick sticky mucus, and it also affect abdominal symptoms, typically pancreatic insufficiency and liver disease. And then there is decrease in digestive enzyme, where there will be steatorrhea or protein malabsorption because there is decrease in pancreatic secretion. So the second type of autosomal recessive disorder is sickle cell disease. It is involved in mutation of beta globin. At least one of the beta globin subunits in hemoglobin is replaced with hemoglobin S. So the mutated hemoglobin tend to polymerize to sickle shape when deoxygenated. So it is typically that the blood that enter the microvasculation, when it is first does not polymerize because there is presence of oxygen, but in the musculature, when the oxygen is extracted, there will be a polymerization of the hemoglobin, and this lead to fragmentation and crystallization of the red blood cell, and thus that will lead to intravascular hemolysis and also occlude the microvasculation. So there will be infarction and pain. So it is typically prevalent in area where malaria is endemic. So the figure shows how polymerized HBS can cause disease. It is typically exacerbated by ischemia, stress, acidosis, and then when that's happened, the oxygenated erythrocyte will be polymerized, and there will be sickle cell formation, and then occlude the microvascularization that will cause hypoxia, infarction, necrosis of vital organ, and also cause pain. And the hemolysis that occur will increase the bilirubin and cause the gallstone jaundice or cholestatic jaundice. And then it also cause vasculopathy and endothelial dysfunction, which later cause hypercoagulopathy and inflammation. So this is also a diagram which explains the pathophysiology of sickle cell disease. And that's all from me. This is my reference, and thank you.
Okay, good. Any questions? Any question on Mendelian inheritance, autosomal dominant recessive? Can you just touch on autosomal dominant and recessive kan?
Yes.
Any questions? Okay, so this one. Okay, so basically in general, this is a very simple concept, dominance and recessive. But apparently, most of the genetic condition, at least 60% of genetic problems are not. I mean, it's not a Mendelian inheritance punya condition lah. But the most common that we usually seen is actually the Mendelian inheritance, the autosomal dominant recessive. So, but you have to understand, as Afiq correctly mentioned, about the difference between penetrance and also expression. Okay, this is things that we have to consider. For example, like NF lah. NF is quite common lah. You'll see NF in your life. Definitely you'll see NF lah. I've seen quite a number of NF. So in neurofibromatosis, for example, sometimes the parents may not be that obvious, okay, or the child might not be that obvious, but they've got it. I mean, if you do like genetic testing, they've got the condition, and if the mother got the condition, if mother or father got the condition, you can see it's almost definitely at least 50% of the children will have the condition. Okay, the penetration is 100%. It will definitely penetrate. However, the expression might be different. Okay, and okay, talk about NF kan. The NF, you have to understand, the first time you encounter a patient where the parents got NF, you may not detect the NF because the presentation of each sign is different. Okay, for example, cafe-au-lait may not be present at birth. Okay, it may be present later, and you may need to use the Woods light to see it, especially those with like darker skin kan. Those with darker skin, you may need to use Wood light, benda macam tu lah. So each of the sign and symptoms may not occur very early on. For example, like Marfan kan, a lot of time children with Marfan, the initial presentation is actually pneumothorax. So they come with pneumothorax, spontaneous pneumothorax. Then you look at the eye, you look at the heart, then that's only the way that you're able to detect Marfan. And a lot of these autosomal dominant conditions are de novo mutation. For example, the parents might not have it. They may be the first in the family to have this. Okay, to have the condition. Okay, what else? And you correctly mentioned, okay, usually the AR autosomal recessive conditions is more severe than the AD. Okay, usually. Okay, the AR is more severe than the AD. For example, typical example is polycystic kidney disease. Okay, PKD kan. You've got two type of PKD, the AR and the AD PKD. Okay, The ARPKD, they presented earlier. They presented earlier. They usually got bilateral disease, and they also got associated with berry aneurysm, liver, polycystic liver disease, and their prognosis is much poorer. Okay, rather than AD. Even though the AD got higher penetrance, more percentage of the children, offspring will get the disease, but most AD, polycystic kidney disease, they are diagnosed incidentally through ultrasound also, or through physical examination. You can palpate the kidney. Okay, and then AR, you correctly mentioned, is most of the time is cause enzyme punya problem lah, enzyme and also function. Enzyme problems in the AR. So that's why most of the IEM is either autosomal recessive or sex-linked. Okay. I think there's no IEM with autosomal dominant. Tapi you've got IEM with sex-linked, IEM with due to AR with AR punya inheritance, and also with mitochondrial punya inheritance. Okay, I think that's all quite straightforward. But kita rest for maybe 5-10 minutes boleh tak?
Boleh.
We'll be back in 10 past 8 ke?
Okay.
Okay.
Doktor sekarang pukul 9, 10 past 8 min.
Assalamualaikum guys. Nak tanya nampak slide tak?
Nampak.
Alright, thank you.
Nampak.
Assalamualaikum.
Waalaikumsalam warahmatullahi.
Okay, Azif ready?
Ready Dr. Okay. Bismillahirrahmanirrahim. So Assalamualaikum warahmatullahi wabarakatuh. So today I will share with you some knowledge regarding sex-linked disorder. So everyone can see my slide? Doctor,
Yes.
okay. So these are my outlines: sex chromosome, sex-linked pattern of inheritance, sex-linked disorder, X-linked disorder, Y-linked disorder, and summary reference. For the sex chromosome, human chromosome usually has 23 pairs, 22 pairs of autosome plus 1 pair of sex chromosome. So my part will specifically explain about the sex chromosome. Okay, the function of the chromosome as mentioned by Sister Fatin Nabiha. So it carries the genes which will portray the traits and character of male and female or female. So for the sex-linked pattern of inheritance, we use Mendelian pattern of inheritance. They are divided into three. The first one is law of segregation, where the dominance and the recessive is segregated. So law of independent assortment, where these two are combined together and there's cross linkage of the genes occur, and it brings up to formation of the daughter, which is under law of dominance, where two different alleles are present. Only one with the dominant features will be expressed. Okay, and then for the sex link referred to gene located at one sex chromosome, it means that the gene is located at the X or Y chromosome. So these are the differences between X and Y link inheritance. I already make it simple. I put it in table so you can refer it later. So for the X-linked inheritance, it is a transmission of gene which is located at the X chromosome. Therefore, if the father is the one who affected, he will transmit his disease to the daughter. And both gender are affected as both gender has X chromosome, and son only receive X links from mother. And for the Y-linked inheritance, it is a transmission of gene located in the Y chromosome. Therefore, the affected father can give his genes, his Y chromosome to his sons. Therefore, the transmission of the disease only occur from male to male. Next is sex-linked disorder. Sex-linked disorder is a genetic mutation caused by a mutation in the single gene located in the sex chromosome, either X or Y. If the abnormal gene is located in the X chromosome, so it is considered as X-linked disorder. While if the abnormal gene is located in the Y chromosome, therefore it is considered as Y-linked disorder. X-linked disorder is further divided into dominant and recessive. So dominant, in other way, you can think of major genes, and for the recessive is minor genes. Okay, and then for the X-linked disorder, female has XX chromosome while male has XY chromosome. Female can be homozygous, which both of the alleles being affected with abnormal gene, and heterozygous, which only one allele is being affected. So if the allele is recessive, so the female will become a carrier, while if the one gene is a dominance, so the female will portray the disease. And males can only be hemizygous because male has only one X chromosome, no carrier form in male. For the X-linked dominant disorder, it affect, it already mentioned in the statement already. So X-linked means that it is abnormal gene located in the X chromosome, and the dominant means that the main gene is affected. So it can affect both gender, male and female, because both have X chromosome. Severe manifestation in male than female. Female is more commonly affected than male. So the examples of this X-linked dominant disorder are hypophosphatemic rickets, incontinentia pigmenti, and Alport syndrome. From the picture on your left, if the affected one is the father, so the father will give his genes to the daughter only. So 100% of the daughter will be affected, while 100% of the sons will not be affected. If the affected mother who carries one dominant genes married with normal father, so the mother will transmit the disease to her daughter and sons. So from the picture, 25% of the daughter will be affected, 25% of the son will be affected, while the other 50% of the children will not be affected. So next we move to the examples. The first example of the X-linked dominance disorder, which is hypophosphatemic ricket. Hypophosphatemic ricket is a type of ricket which characterized by low phosphate level in the blood due to resistance to vitamin D. It has an abnormal problem in the intestine where he cannot absorb the calcium. Therefore, the level of the calcium in blood will be reduced. The body will will detect these abnormalities and increase the release of the PTH hormone from the parathyroid gland. Parathyroid hormone, the function of the parathyroid hormone is to increase back the calcium level by increase the absorption, and also it will increase the excretion of the phosphate that can lead to hypophosphatemia. Okay, these are the features of the patient with hypophosphatemic rickets disease. So as you can see, the patient will have short stature, pigeon chest, spine deformities, pelvic deformities, and bow leg. The most obvious thing that you can see is bow leg. This is due to bone growth deformities. Okay, and then next is the incontinentia pigmenti. Incontinentia pigmenti is the neurocutaneous disease due to genetic disorder problem. It is characterized by features of problem with the neurological problem, the cutaneous problem, and also the dental problem. It is commonly affected in female at early age, and usually in infants, the patient will portray blistering skin rash with wart, W-A-R-T, like growth. Okay, and as the infants grows into a children, so he or she will manifest hyperpigmented skin and that will further fade when he or she reach adolescence. Okay, next is the Alport syndrome. Alport syndrome is X-linked dominance disorder which due to mutation in COL4A5 gene. It is characterized by progressive loss of kidney function. If it is not treated early, it can lead to end-stage renal disease, and it also can affect the inner ear, which can lead to hearing loss. Alport syndrome can also affect the eye by damage the layer of the retina, the retinal cells, and that can lead to loss of vision. And the presence of the hematuria usually is related with the Alport syndrome due to progressive kidney dysfunction. Okay, and then for the X-linked recessive, to make it simple, if there is presence of one recessive gene in the male, so he can present, he will manifest the symptoms. While if one recessive gene is present in female, so she will become a carrier. Okay, at least two genes are required to portray the disease. This is called as homozygous in female. Okay, so male is commonly affected than female, and the symptoms are more severe in female. Female carrier may have mild symptoms. So if the patient, the female carrier may have mild symptoms, that is not very severe with the patient that manifests the symptoms. with dominant genes. Okay, examples of X-linked recessive are hemophilia, G6PD deficiency, Duchenne muscular dystrophy, and color blindness, especially red-green. Okay, based on the picture, on your left, if the affected one is the father, so father will give his genes to the daughter. So 100% of the daughter will become carrier and 100% of the son will not be affected. If unaffected father married with a carrier mother, so this mother has one recessive gene in his in her allele. So 25% of the daughter will become a carrier, 25% of the son will be affected, and 50% of the children will not be affected. If the affected mother carries two recessive genes on her chromosome, therefore the children, especially the daughter, will become carrier, and the son will portray the symptoms. Okay, the example of X-linked recessive, the first one is the most common one is hemophilia. Okay, there are two types of hemophilia, hemophilia A and hemophilia B. Due to a genetic mutation, therefore, it will leads to factor 8 deficiency for hemophilia A and factor 9 deficiency for hemophilia B. From the picture, you can see that hemophilia A is more common compared to hemophilia B. Okay, from on your right side, I put diagram of coagulation cascade. From the coagulation cascade, factor 8 and factor 9 are important in intrinsic pathway, which is important to convert the prothrombin into thrombin. Okay, this thrombin activation will convert the fibrinogen into fibrin, and which can lead to cross linkage of the fibrin, leads to blood clot. If there is deficiency in factor 8 and factor 9, therefore there will be problem in the blood clotting and leads to prolonged bleeding. So the clinical features are recurrent spontaneous bleeding into joints and muscle, which can lead to crippling arthritis. Intracranial hemorrhage, bleeding post-circumcision is a common question that you can ask to the children if you suspect hemophilia. Prolonged oozing from the venipuncture site. Okay, from the picture, causes of the hemophilia, if the father married with carrier mother, okay, we'll continue. So if the father married with a carrier mother, so the mother will pass her disease to daughter and son. Therefore, 25% of the daughter will become a carrier and 25% of the son will be affected, while the other 50% of the children will not be affected. If the affected one,
I think it's actually 50% of the sun will be affected.
50%.
Because if you look at the sun, 50% of the sun will be affected.
Okay.
25% of the daughter, 50% of the children will be okay lah. 25% of daughter will be affected. Tapi for X-linked is 50% of the son. Kalau mother of the son lah, bukan of the children, of the son.
Okay, understand that. Okay, sorry. So 50% of the son will be affected, okay, while the other daughter will not be affected. If the affected one is the father, married with a normal mother, therefore 100% of the daughter will become carrier and 100% of the son will not be affected, as the father cannot transmit the disease into his son. And then for the G6PD deficiency, G6PD is a important enzyme for HMP shunt, which is HMP shunt stands for hexose monophosphate pathway. Okay, while G6PD stands for glucose 6-phosphate dehydrogenase. This G6PD enzyme is important in conversion of the glucose 6-phosphate to 6-phosphogluconate, and then it will release the NADPH. This NADPH will then oxidize the glutathione, and the glutathione, the importance of the glutathione is to protect HB oxidation, prevent the hemoglobin from hemolyze. Sorry. Okay, therefore, if there's presence of this SPD deficiency, the oxidation of the glutathione will be reduced, and it leads to HB oxidation where it can hemolyze. Okay, if there is the factor is if there is presence of external trigger. Okay, if there is no external trigger, so the patient will become normal, will be like normal person. Okay, so the patient with G6PD deficiency, if there is presence of external triggers such as drug, acetaminophen, sulfonamide, and also stress, infection, and also the food such as fava bean, so he or she can manifest the symptoms of jaundice, dark color urine, back pain, and anemic symptoms. So investigation that you can do in this patient is full blood count. In the full blood count, you can see that they will be increased in the erythropoiesis. And the second one, there will be presence of the bile cells and hence body in the full blood picture. So the treatment for this patient is you need to avoid the external trigger. Next is Duchenne muscular dystrophy. It is a genetic disorder in childhood. It affects 1 in 3,500 boys, resulting in mutation of the DND gene. It is characterized by degenerative, muscular degenerative, and also weakness in this patient. The clinical presentation for this patient is hypotonia, weakness, heart problems such as cardiomyopathy, vertebral problems, scoliosis, respiratory decline, cognitive and behavioral dysfunction. As you can see from the features, from the picture, the features of the Duchenne muscular dystrophy patient mostly affected the muscle part, the part of the muscle. Therefore, these are the features. The shoulder and the arm are held awkward, back awkwardly when walking, belly stick out due to weak belly muscles, poor balance because of the muscle. So the patient may fall often, awkward clumsiness in walking, thick lower leg muscle, and also weak muscle in front of the leg cause foot drop and tiptoe contracture. Usually it affect the muscles in this patient. Next is color blindness, especially red-green, where the patient cannot differentiate between the red and green color. Cannot see the red and green color. So any one of you knows any test that can be done in patient to detect color blindness? So the answer is Ishihara test. So thank you to me. Okay, next is Y-link disorder. It is a male-to-male transmission because only male has Y chromosome. It related to male sex determination and reproduction. So it affects the reproduction. So leads to infertility in male. Okay, the examples are male infertility, auricular hypertrichosis, and also retinitis pigmentosa. For the first examples, auricular hypertrichosis, it is also known as werewolf syndrome, where it is characterized by hair growth anywhere on the body. There is no known cure for this disease, but you can ask the patient to shave to maybe can minimal the presentation. Okay, and it is a rare disorder. Next is retinitis pigmentosa. It involves in breakdown and loss of the cells of the retina. So the patient will have a problem at night to see at night, and also problem with the peripheral vision. So the patient can only see on the central, have central vision only. So it also considered as a rare disorder in this patient. Okay, next is the last one is summary. So I already make it simple. I put it in table so that you can see it later. The differences of the sex-linked disorder. For the X-linked dominant, the transmission, there will be no transmission from father to sons because the son will only receive Y chromosome from the father, and X-linked recessive also the same. For the Y-link, the transmission only from father to son, so male to male transmission. The carrier, next is carrier or affected gender. The female is not affected. There is no female carrier in the X-linked dominant. Okay, because if there is presence of dominant gene, okay, at least one gene, it is already can portray the disease. Okay, for the X-linked recessive, the female require two recessive gene to portray the disease. If the female, if there is presence of one recessive gene in the chromosome of the female, so she will become a carrier. And for the Y-link, only male are affected. Okay, the incident by gender comparison, female are more common than males in X-linked dominant, while male are more common than female in X-linked recessive. For Y-link, only male are affected because it related with Y chromosome. Okay, for the commonest, X-linked dominant is less common compared to X-linked recessive, and the Y-linked disorder is very rare. So I also put differences between autosomal and sex-linked disorder. For autosomal disorder, it appears in both sexes equally. The sex-linked disorder, it appears more common in males. For the autosomal disorder, it affected alleles that can be inherited from either parents, while in sex-linked disorder, sons only receive the affected X link from the mother, and the daughter receive the X link from either parents, either father or mother. So that's all for me. These are my references. Thank you.
Okay, good. Any questions?
I have a question.
Okay, good.
I want to ask regarding the X dominant disorder. I want to ask about Fragile X syndrome, though you didn't give that as an example, but I've read that in Fragile X syndrome, the unaffected father can transmit the disease to his grandson through his daughter. You got me? How could that happen? How the unaffected father could transmit the gene to his grandson to his through his daughter? That's the question. Thank you.
Isha, can you repeat the question?
The question is how the unaffected father could transmit the disease to his grandson through his daughter. About Fragile X syndrome. Thank you.
Okay.
Okay, thank you, Isha.
Okay. I'm not sure whether Fragile X is actually X-linked dominant. Memang Fragile X is X-linked dominant eh?
Fragile X.
Can someone check? Sure, I thought it's recessive. Let me check first. But anyway, can someone check?
Doktor, X-linked dominant.
X-linked dominant eh? Okay.
Uh.
So actually that's why I think siapa yang tadi presenter, the previous presenter present about the penetrance and also the expression kan. You know, some disease they did not penetrate 100%. The penetrance is not 100%. So they've got skip generation, or they not inherited. It seems like it's not inherited. For example, sometimes you do a pedigree, a 3 or 4 generation pedigree, and suddenly there's loss. This is an dominant. If the for a dominant disease, dominantly inherited disease, you got this we call skip generation. So it may not present in a generation, it just skip. We don't know why, but it just skip. Okay, and also that's why this disease that we call low penetrance disease. Some disease is 100% penetrance. For example, like NF lah. NF definitely you will see, if you do the pedigree, you will see like every generation will have NF. You've got one NF in the family. But in disease that have low penetrance, you may skip the generation. But it may appear again later. It may appear down the line, it may appear. And Fragile X is very common. Fragile X is one of the most common genetic disease you'll meet. And actually, if you found anyone with delay kan, developmental delay or learning disability, one of the things you have to work out is for Fragile X. Okay. Understand kan? Did I answer your question?
Alright, thank you, Doctor.
Okay.
Thank you, Doctor.
Okay, lepas tu what else? For the X-linked dominant, okay, if you draw the pedigree, it's very difficult to distinguish the X-linked dominant than to the autosomal dominant, because it's very similar. Because everyone got the X, and once it's dominant, it doesn't matter. You just need one, or you just need one X, then you'll get it. So the pedigree, if you do a pedigree of X-linked dominant, is similar to the pedigree of autosomal dominant. Okay. And then what else? So disease like I'm sure if you're doing pediatric, I'm sure in your lifetime you will come across incontinentia pigmenti, and also this definitely GSPD. Hemophilia is very common. GSPD is very common. DMD is more common than hemophilia. Then you'll definitely meet one of the DMD at least. So this is not that rare lah. Actually, X-linked disorder is not that rare. You'll meet one of these at least once in your lifetime. And for example, like incontinentia pigmenti, kan, the disease is quite mild. Okay, even though some people have got a severe disease where it affect the kidney lah. Usually the kidney is affected. But baby with incontinentia pigmenti, okay, they born with a skin lesion, very gross looking skin lesion. I've met 2 ke 3 down in my career lah. Very gross looking. You think this is infection, looks like bullous impetigo sikit. Tapi very gross looking lah. But after a while, it resolve. And it leave you with the hyperpigmented. So sometimes baby, if you got a baby, okay, especially female lah, you can get male incontinent pigmented as well. So it's X-linked dominant, tapi usually it's female. So if you see a baby with rashes like the picture kan, it's a race rashes, or it's a popular rashes. And then what you can do is just look at the mother, okay, examine the mother. You'll see the lines, the Blaschko lines on the mother. So this is what you call Blaschko line lah. So you will see the Blaschko line, and it's a neurocutaneous manifestation, because some proportion of these children will grow up with neural problems. They can have epilepsy, they can have learning disability, they can have eye problem and kidney problem. But most of them are normal lah. Cuma some of them can get neuro problem and nephro. Okay, what else? X-linked. Any other questions ke?
Doctor, I have a question about DMD. You just said that gene therapy is a promising treatment to treat DMD. So will it give a excellent prognosis, or it just slow down the progression of the disease?
Actually, there are few children in the world has been treated. Okay, so far it show promising results. Cuma the trick is you have to diagnose very early. Okay, most of these baby undergo the treatment even at embryonal stage pun. Okay. What good about the MD, because it's a single gene disorder. We already know the disorder, the gene, the culprit gene, and the MD is the most extensive research disease in the world, genetic disease. Everyone know everything about the MD, almost everything about the MD we know. So the treatment is quite simple lah. I mean, the concept is quite simple, because it is recessive. So we can just add another copy of the gene into the genome lah. You can just put it in the X chromosome next to the gene lah, next to the recessive gene, and then it will translate it and produce the protein lah, the dystrophin protein. So far, I mean, those successfully treated, most patient yang underwent gene therapy for DMD, I think is successful lah. If I'm not mistaken, it's successful. Those yang berjaya lah, because the main challenge is actually to transfer the plasmid containing gene to the to the cell lah, to the cell. To that's challenge. Once it's there, then you are cure. You won't cure because at the end, mother with DMD, a carrier of DMD, they will have symptoms. But they won't get severe symptoms like Becker or Duchenne. Tapi they will get symptoms. Mother who carry the mutation, they will get they will have some sort of weakness, usually calf pain. And then if you look at the CK of this mother of children with Duchenne is usually more than 1,000. For Duchenne, they'll get like 30,000 lah. CK dia kalau you ambil memang 30,000. For Becker is slightly milder lah. Becker MD usually maybe like CK dia around 10 to 20,000. Tapi mother of carrier of Duchenne gene, they will get mild symptoms. Even though Duchenne itself is a recessive disorder. Okay, and then Duchenne is a proximal. When we talk about muscle-nya pathology kan, either dystrophy ataupun myopathy kan, we can classify into proximal or distal. Okay, and Duchenne is a is a proximal problem. So the problem is actually proximal muscle. That's why they've got shoulder arm problem. They've got the thigh. They've got weakness actually at the thigh tau. It's not at the distal. That's why they compensate with calf hypertrophy. So the early sign of Duchenne is they've got the Gower sign kan. Gower sign because they've got weakness, proximal weakness, so they cannot actually they need to stand using their distal muscle, especially the arm and also the distal leg. But as a compensatory mechanism, they've got hypertrophy. So initially they've got hypertrophy of the calf muscle, but later on the hypertrophic muscle will be substituted by fat. So they become hypertrophic, but it's fat rather than muscle. And later all the muscle will degrade and change to replaced by fibrous and also fat tissue. It's very sad, no? DMD is a very sad disease. But I've got a disease which has much what you call it? Sad lah, which is the Huntington. I'm not sure whether any of you seen Huntington. Ada pernah jumpa Huntington kat Malaysia?
No.
Huntington is a very sad disease. Kita ada banyak. I've met a lot of Huntington. Huntington is quite common. It's affected, I think, 1 in, maybe more than Duchenne kot. Maybe around there lah, 1 in 5,000 macam tu. Tapi biasa banyak dekat European country lah. But I've met a lot of them and it's very sad. Nanti habis ni cerita pasal kenapa Huntington is a sad story lah. It's a sad disease. Tapi later, any other questions? Ada? Ada questions? Tak ada?
No.
Okay, tak ada, then we move to the last presentation lah.
Thank you, Doctor.
Okay, hopefully we can finish by 10:15 ke, 10:10 ke. Okay, next. Actually kan, you guys kena buka video tau, everyone, sebab this is how they check your attendance. Because for your, I'm not sure whether you're aware, for your assessment for this semester kan, the main assessment is actually from class kan, from class, and also kita pakai this non-formative assessment, which is your presentation, your long case, short case classes lah, from class. We assess you from class. I think up to 30% kan, macam tu lah, pendek kan sangat. Tapi sebab because we won't have any long case, tak ada rasanya exam. Long case tak ada. Tak ingat nantilah. Tapi you get a lot of point from the class. Okay. Continue.
Yes. Can everyone hear?
Yes.
So now let's learn about chromosomal disorder. So as we know, everyone have 23 pairs of chromosome, 22 pairs of autosomal chromosome, and a pair of sex chromosomes. So chromosomal abnormality results from alterations on either the number of the chromosome or the structure of the chromosome. Okay. So let's go to the first classification, which is the autosomal chromosome, which is Down syndrome or trisomy 21. It is the most common autosomal trisomy and the most common genetic cause of severe learning difficulties. So the overall incidence is about 1 per 650 live birth, but the individual incidence is dependent on the maternal age. The the risk of having child with Down syndrome increases with the maternal age. However, most Down syndrome babies are born to younger mothers because proportionately younger mothers have more babies. Okay, so let's go to the cytogenetics of Down syndrome. The extra chromosome 21 may result from meiotic non-disjunction, translocation, or mosaicism. So in meiotic non-disjunction, it happens when a pair of chromosome 21 fail to separate, this one, and so that one gamete has 2 chromosomes and one have none. So when the child is born to the take the extra gamete, so he or she will born with Down syndrome. Next, in Robertsonian translocation, the extra chromosome 21 is translocated into usually chromosome 14 and in some cases to chromosome 15, as you see here. And the last one in mosaicism. Mosaicism results from non-disjunction during mitosis after fertilization. Therefore, here some cells will have a normal. Some cells will have normal chromosome, which is a pair of chromosome 21, and some cells will have 3 chromosome 21, and this is the milder phenotype of Down syndrome because not every cells have 3 chromosome 21. Okay, the clinical features of Down syndrome, as you can see here. Okay. The facial appearance. Babies with Down syndrome usually have round face and short neck, flat occiput, which or brachycephaly, hypothyroidism. Okay. Hypothyroidism, which is the abnormal ratio between interpapillary distance and the distance between lateral canthers. And then they will have bilateral upward slanting of eyes, congenital cataract, squint or glaucoma, epicanthic fold, flat nasal bridge, protruding tongue, and low set ears. For the upper and lower limbs, they will have a single palmar crease, which is also called simian crease, clinodactyly, which is incurved fifth finger, short stubby fingers, hypotonia, and cellular gap between big toe and second toe. For the cardiovascular system, some of them will have congenital heart defects. The most common one is AVSD, and then followed by VSD, PDA, and ASD. Then for the gastrointestinal system, they will have feeding problem due to the hypotonia, which results in poor feeding due to poor suckling. And then duodenal atresia, pyloric stenosis, and tracheoesophageal fistula. For the and also Hirschsprung disease. For the central nervous system, some will have congenital cataracts and glaucoma. And for the endocrine system, it also includes hypothyroidism and hypogonadism. And for musculoskeletal system, okay, some will have congenital hips dislocation and lentoaxial instability. For the during infant and childhood, boys with Down syndrome is usually infertile, and girl menarche is like only slightly delayed. During adult life, they will have increased risk of dementia and shorter life expectancy. For the diagnosis of Down syndrome, it can be diagnosed antenatally or but usually most of them is diagnosed during birth because of the features of Down syndrome. So for the antenatal diagnosis, we can do ultrasound, amniocentesis, or chorionic villus sampling. For the ultrasound, we can check for the knuckle fold thickness. They will have a knuckle fold thickness more than 3 millimeter at 10 to 14th week. And for the we check for the double bubble sign, which indicates dilatation of proximal duodenum and stomach. We can also do amniocentesis to and then do karyotyping to check for the extra chromosome, and also fluorescence in situ hybridization. For the chorionic villus sampling, which we can do triple assessment, which is which results in low AFP, low estradiol, and high HCG. And for the newborn, the diagnosis of Down syndrome is usually we do karyotyping or fluorescence in situ hybridization. For the management of Down syndrome, the following information and explanation should be conveyed to the parents before discharge from the hospital. The first one is the information on Down syndrome, short and long-term complications of Down syndrome, counseling to deal with a feeling of disappointment, anger, and guilt, contact with local parent support group, and advice on potential recurrence on risk and contraception until chromosome diagnosis is available. For the examinations on the complications of Down syndrome, for the growth, appropriate growth monitoring is essential. Those who are excessively short or underweight may have additional pathology. Usually, those who have failure to thrive usually have underlying cardiovascular pathology. And specific growth chart, which is a Down syndrome growth chart, is used in Down syndrome patients. They don't use a normal growth chart for. They don't use the growth chart for the normal babies. And for the examination of the thyroid gland, baseline T4 or TSH at birth must be obtained, and all patients with Down syndrome must have cardiac status, and the cardiac status must be established by the age of 6 weeks. For the cervical spine instability, people with Down syndrome have small risk for major neurological damage caused by cervical spine instability. So, cervical spine X-ray is suggested, and the symptoms of spinal cord compression includes the neck pain, unusual posturing, and change in gait. For the hearing and ophthalmology, hearing assessment must be done in patients with Down syndrome, and the aim is to establish whether the permanent hearing loss is if there is permanent hearing loss by the age of 10 months. For the examinations of OSA, obstructive sleep apnea, most some patients with Down syndrome may have OSA because they are hypotonia, right? So they are the tongue will be more susceptible to block the respiratory system during sleep. So and this is a major contributor to pulmonary hypertension. So ENT referral is must be done. The next the second most common trisomy in children is Edwards syndrome. So Edwards syndrome is the second most common trisomy. It occurs in 1 in 7,500 live birth, and most of them are female. It results from the extra chromosome located at the chromosome 18. Most of them have low survival rate, and more than 95% spontaneously aborted in the first trimester. The clinical features of Edwards syndrome include small and premature appearance, hypertonia, prominent occiput, micrognathia, which is or mandibular hypoplasia, which is a condition in which a child has very small lower jaw, short sternum, and clenched hand. As you can see here, different from the Down syndrome patient, which is usually have a flat occiput, but in Edwards syndrome, they usually have a prominent occiput. The third most common trisomy is trisomy 13, which is also called Patau syndrome. It results from the extra chromosome located at the chromosome number 13. Patau syndrome children, it is usually fatal in the first year of life, and less than 10% of patients survive beyond 1 year of life. And they will have severe intellectual disability and physical abnormality. Okay. So the clinical features include small head, absent eyebrows, cleft palate, malformed ears, clenched hands, and cyclopia, which is a single or partially divided eye existing in a single orbit. And then they have club feet or rocker bottom feet, which is characterized by a prominent heel and a convexly rounded zone. Some will have aplasia cutis congenita, which is absence of skin. This one, absence of skin, with or without the absence or underlying structures such as bone. It is most commonly affects the scalp, but any location of the body can be affected. Next is Klinefelter syndrome. Klinefelter syndrome. Klinefelter syndrome results from the extra X chromosome. They will have two X chromosome and one Y chromosome. Before puberty, boys with Klinefelter syndrome are phenotypically indistinguishable, but the diagnosis often made when the boy is 15 or 16 years old when they reach puberty. So the clinical features include tall stature, slightly feminized physique because they have extra X chromosome, frontal no frontal baldness, poor beard growth, breast development, osteoporosis, female type pubic hair pattern, and small testes. For the treatment of Klinefelter syndrome, we can do androgen therapy is used to correct the androgen deficiency and to provide appropriate virilization, which is to promote hair growth and to improve psychosocial status. And before this, Klinefelter syndrome is categorized under infertility, but now now it is found that almost 50% sperm of Klinefelter syndrome patients are viable. So we can retrieve the fresh sperm, which is viable, and do IVF technique. The next chromosomal disorder is 45XO, which is Turner syndrome. Turner syndrome results from the absence of X chromosome. So they will only have one sex chromosome, which is X. Okay. The clinical features include short stature, characteristic facial features, low hairline, full of skin, cardiovascular disease such as constriction of aorta, poor breast development results from the absence of one X chromosome, widely spaced nipples, small fingernails, elbow deformity, rudimentary ovaries, and some will have no menstruation, and presence of brown spots around the body. For the management of Turner syndrome, height monitoring, cardiac evaluation, BP measurement, echo at baseline, and every year is recommended for the to check for any cardiovascular pathologies. Growth hormone therapy because they will have short stature, and ovarian hormone replacement to elicit increase in height and development of secondary sex characteristics. The next classification of chromosomal abnormalities is the structural abnormalities. So one of them is Cre-Duchat syndrome. It is also called 5p minus syndrome. It results from a deletion in the short arm of chromosome 5. The deletion of chromosome 5 is usually inherited from the father. It is very rare. So it is babies presented with babies who have Cre-Duchat syndrome will present with high-pitched cry. It is characterized by cat-like cry during early infancy. This unusual cry is due to the structural abnormalities of the larynx. Example, laryngeal hypoplasia and CNS dysfunction. The laryngeal appearance may be normal or may exhibit marked anatomical abnormalities such as floppy epiglottitis, small larynx, and asymmetric vocal cord. About 1/3 of the children no longer exhibit the cat-like cry by the age of 2 years. And the clinical severity of the Cre-Duchat syndrome depends on the size of the chromosomal deletion. The larger deletion on the chromosome 5 will result in more severe expression of the disease. So the clinical features include cardiovascular congenital heart disease, hypotonia, facial appearance such as microcephaly, hypertelorism, and low set ears, slow swallowing difficulty due to the hypotonia, and developmental disability. And the last syndrome in in the chromosomal abnormalities is 15 chromosome 15 deletion, which is Prader-Willi syndrome. So the clinical features, it results from the deletion or unexpressed maternal chromosome 15. It is very rare, and the characteristic features are hypotonia, short stature, chronic hunger, incomplete sexual development, cognitive disability, and behavioral problem. So this is the during infancy, they will have poor feeding due to the hypotonia, short stature, and delayed puberty during childhood. They will have obesity during childhood, but poor feeding during infancy. Okay, that's all. Any question?
Okay. Assalamualaikum. So just start work. Okay. Okay. So you touch on most of the important chromosomal disorder. Prader-Willi. Oh, you mentioned about Prader-Willi. Good, good, good. Prader-Willi is a special group, right? It's a special group. Did you? Okay. So basically, can you go through your slides, kejap? Go up the slides. Okay. Ini kerja cak kan lagi. Ni bukan ni. Boleh. Lagi. Got trisomies. Got the monosomies kan. Kena filter. Video chat. Lagi. Lagi, last kali. Prader-Willi.
Prader-Willi.
Okay, so good. So almost all you presented. Yes, of course, the most common is Down syndrome, and then you've got the Edward and Patau in sequence. So in general, Edward and Patau, we don't do active management lah in general. If they alive, then we support, but we don't aggressively treat. But for Down syndrome, they are considered a subtype of normal human. So it means that we have to treat them as human, which means we have to actively treat whatever deficient they've got. And then you've correctly mentioned most of Down syndrome is diagnosed postnatally. However, actually the main reason is not that because we cannot diagnose antenatally, but because it won't change anything. Okay, in other countries, in developed countries, for example, in the Western countries, Down syndrome is diagnosed antenatally through CVS or amniocentesis kan. Because they've got the option of termination. Okay, they can do TOP lah. But in Malaysia, because we don't do TOP, and most people won't agree doing TOP, and it's illegal for Down syndrome. It's illegal to do TOP. So even though the diagnosis is mainly for preparation, it's not for management. So parents, we do offer parents. For example, if we do detail scan, we think there's a like thickened knuckle thickening or fat pad, some features that might suggest Down syndrome. We can offer the parents for antenatal diagnosis, antenatal screening. If it's turned out to be positive, then it's more to a preparation lah for them to prepare physically, mentally, financially, semua lah. But they've got no option to terminate for Down syndrome. But for others, they can. Okay, according to Majlis Fatwa, Majlis Fatwa allowed, and even the KKM allow for TOP for some diseases. For example, thalassemia. Okay, thalassemia, if both parents are carrier, especially beta major, and then beta carrier, and then the baby, the embryo confirmed to have beta major, then is allowed to TOP. Hemophilia, for example, is allowed for TOP. Even though these conditions are actually, there's a lot of ethical issue regarding genetic, and this is very interesting issues and very real issues. It's not something that theoretical. So things like, even though hemophilia, they can live like normal people, they've got normal intelligence, fertility no issue, life expectancy of hemophilia is marvelous, but yet because in future we cannot support hemophilia, so that's why it is allowed for TOP for hemophilia, because cost of treatment for one hemophilia boy is at least 1 million per year, and nowadays they can only get treatment from KKM. So when we decide for TOP, for decision whether to actively treat or to stop treatment, to withhold treatment, a lot of things we have to consider. Okay, not only because not only the disease, we not only talk about quality of life, we not only talk about function, we also talk about quality of life. Kan, contoh macam Duchenne kan, if you accidentally diagnose Duchenne, you can actually terminate. Okay, because they later on they have a lot of, Duchenne children, they've got normal quality of life, but functionally, this a sad disease, sad disease. And then so these are the things that we have to consider. And then, so you correctly mentioned about the trisomy. We've got the 3 trisomies. So Edward and Patau, both are conservative treatment. Bear in mind, usually Down syndrome can easily diagnose clinically, but Edward, it may not so. Edward might be a bit tricky. But in general, Edward, they are usually late preterm baby. So they born quite early at 35, 36 gestational age, and they are very cute. They are very cute. If you see a cute baby, small, they usually SGA. So small late preterm baby, very cute, but something not right with the baby. Then most of the time is Edward. Okay, rather than Patau. Patau is gross lah. You can diagnose Patau very easily. They've got cleft lip, cleft palate. They've got obvious, like sometimes they're born with single eye, cyclops. So Patau is quite obvious, but Edward is slightly difficult to diagnose. And most of the time, there's something wrong with the baby, but they are cute and small, and they born late preterm. And they've got these overlapping fingers, rocker bottom, cutis aplasia. So, and they've got very small pelvis. Okay, so the knee is like knocking, and the hip is very small. So these are features of Edward. And then you've mentioned about Klinefelter. Klinefelter, most of the time you diagnose at puberty. As you correctly mentioned, at baby is very difficult. Turner, a lot of Turner can be diagnosed if they fortunately born at tertiary center with specialists, then Turner is quite easy to diagnose. Okay, in a year you diagnose just as many Turner as Down syndrome, maybe slightly less, just slightly less Turner than Down syndrome, but you meet Turner, there's a lot of Turner out there. So the clinical features at birth, they've got these edematous periphery, edematous subcutaneous edema over the feet and the palm and the hand, and then they've got this web neck. Later on, they got short stature, all the features that you've shown lah. Turner, most of them, I think all of them got slightly low IQ, but slightly not that bad. Slightly low IQ, they're infertile, and the main issue with them is short stature. Okay, that's why they need growth hormone. Okay, we need to give growth hormone, and then you need to give hormone replacement to induce ovulation. Okay, because if not, they won't develop their secondary sexual characteristic. Okay, and then what else? Klinefelter, Turner. Okay, Kliduchat. Even though you've mentioned it's quite rare, I've seen a family of Kliduchat. Actually, Kliduchat, this one of the disease, one of the conditions that always come in exams, especially postgraduate exam. Because it's, even though it's mentioned, it is rare, but it's something that you have to detect. I've got a family of Kliduchat because of stable translocation, where actually the parents, the mother family carried stable translocation 5 and 8. So which means the 5th chromosome is translocated on the 8th chromosome. So the baby born, the family, I mean my patient's family, and also actually all the families born in USM. So this family, they've got 2 affected daughters. One passed away, one still alive. Oh no, both passed away. And then the cousin of the mother's cousin also got 3 siblings. Got 3 children. 3 affected, 1 passed away, 2 still alive. They are quite okay lah. So in this family, what happened is they've got stable translocation of 5 on 8. So the 5th chromosome translocate on the 8th chromosome. And what happened is the child either get normal, they are either normal, or they've got monosomy 5 or trisomy 5, or monosomy 8 or trisomy 8, or normal. So these are the outcome of the children. So apparently my patient's family, my patient's siblings, and my patient's children both got monosomy 5, which is the worst lah. So the worst form of Kliduchat. So both passed away during quite early natal period. But her cousin, most of the child got trisomy 5, which is I think it's the opposite of Kliduchat. So they've got gross deformity as well, but they alive. So, and also I know someone very close to me with Edward, stable translocation Edward. So like the trisomy, the causes can be divided into 3: the non-disjunction, the mosaic, and also the translocation kan, Robertsonian, especially Robertsonian translocation. So Robertsonian translocation, people who carry the Robertsonian translocation, they are normal, but their children can get the trisomy. Okay, kan? Okay, these are usually young children, young couple, and maybe that more than one of the children get trisomy. Okay, and then you've mentioned about the Prader-Willi. This is very interesting. This is a concept of genetic imprinting and lionization. So this is something different because in Prader-Willi, what happened? You've got I think chromosome 5 kan? 5 ke 15 15 I think. So what happened? 15 deletion. So what happened is the child with Prader-Willi, they receive two short arm 15. I think short arm 15, 2 short arm 15 from the mother. So they've got loss of father's punya chromosome. So they've got 2 chromosome 15 from mother. So they've got Prader-Willi, or they've got the father's. They've got one from mother, one from father. But something happened to the father, so it's we call imprinted. So means the fathers become inactive. So when the father's chromosome become inactive, it seems like the mother, the child only got one from mother, so it become this is what we call genomic imprinting. So they've got Prader-Willi. The sister of Prader-Willi is called Angelman syndrome. So if the same thing happened, but the child got from mother instead of from father, so they will present it as Angelman syndrome. Okay, just imagine for chromosome 15, everyone, I mean the child will get one from mother, one from father, isn't it? So sometimes what happens is they've got 2 from mother and none from father because of translocation at the father's gamete, leading to the child got 2 from mother and none from father, so they become Prader-Willi. But sometimes what happens is, they get 2 from father and none from mother, then they become Angelman syndrome. Okay. Okay, so this Prader-Willi is slightly ni lah. But you see Prader-Willi. Prader-Willi is not that uncommon. So I've got like 1, 2, 3. I think I've got 2 or 3 patient Prader-Willi, and they've got ferocious appetite. They've got problem with metabolic disease because they eat a lot. So they're very obese and they die very young lah. They die very young. Okay. Any other questions?
Doctor,
Huh.
do you know any other condition where TOP is approved by fatwa?
The one that I'm not sure. There's no extensive list. Okay, there's no. Usually, it based on a individual cases cases by cases but for hemophilia that we are sure of is hemophilia, beta major, thalassemia major, Edward Patau, some Duchat also I think is allowed. There's no extensive list. I'm not sure if there is, but these are some of it. Prader-Willi tak sure. Prader-Willi tak ada kot dalam list. It's quite rare. Tapi Edward Patau memang confirm. Thalassemia and also hemophilia is confirmed. Recently we've got issue with Zika virus kan, where the Mufti said can terminate. But apparently Zika virus, last time we thought Zika virus leads to microcephaly and so some other presentation, but it's actually not. It's not true. Okay, it's not evidence based. The latest evidence said microcephaly is not associated with Zika. Okay, ada lagi? Tak ada soalan dah? So genetic is one of the topic that I'm really interested in, and I'm sure if you read around genetic and if you meet more genetic condition, then you will be able to appreciate the nature, the beauty of genetic diseases. Okay lah, so rasa tu cukup. So any last questions before we stop?
No, Doctor.
Okay, if no, then kita tangguh hantar speaker forum. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Doctor.
Okay, thank you.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Doctor.
Assalamualaikum. Habis ke kelas ni?
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam.
Okay.
Salam.
So let's begin with Kitab Al-Fatihah. Bismillahirrahmanirrahim. Alhamdulillahirrahmanirrahim. Iyyaka na'budu wa iyyaka nasta'in. Ihdinas siratal mustaqim. Siratal ladzina an'amta 'alaihim ghairil maghdubi 'alaihim waladdallin. Alright. So actually who has duty to present the case?
Saya rasa. But like Alif WhatsApp you this morning about the patient.
Yeah, so we just stop lah to clock the case, but we may discuss the case after this lah. Okay. Maybe it is your first experience. Maybe you have previous experience before about this, but this is live lah. So we have live lesson today. Yeah. The thing that we call it soft skill is not you cannot get the soft skill during your official or formal teaching process. So we learn from life, from your experience, from everyone's experience. So first thing that I want to remind you is our basic requirement as a Muslim kan, after we declare our faith or belief, iman, we do syariah as Muslim, Islam. We should also encourage akhlak. We do our best following the path of Rasulullah SAW. Sometimes we have some bad experience like this. So the 2 points that I want to highlight is first, of course, the empathy kan. So you learn kan, some patients, some parents maybe doesn't or don't want to be what we call it to be
interviewed
or to be diganggu lah kan, to be terganggu lah. Sometimes they feel that when we call the patient, it is not a good time for them. Actually, so that's why we should give some more room space lah to the parents macam ni. If they don't want to be interviewed, just let it be lah. No need to push lah. Okay, so empathy, so feel what they feel. Okay. Alright. Another thing maybe about your patient lah kan. When you contact the mother usually?
I got the number on Friday. So I directly WhatsApp the mother after Jumaat. And then she said she free at night. And then I asked her, is it okay if I do the video call? And then she said she doesn't want the video call. She said just ask through WhatsApp. So at night when I start asking question, he just answered a few of my question, and then she stopped answering at all.
That's why maybe she was busy kan. It probably due to her activity busy, tak sempat ke, atau it is possible also from previous experience, maybe tak tahulah yang previous student yang clerk her tu did something rude ke apa-apa lah. So it is give effect to your process kan. So okay lah. Because previously when I met her many times she was okay. Even to be interviewed lah. So she was okay to meet the students before. Alright. So just stop lah, because if we push her to do further, I'm afraid we have some legal issue here. Even when I give you the contact, it's still illegal because we don't have written consent. Even academic affair pun don't allow actually. If you ask Prof Marzuki, he doesn't allow to do online interview or clerking. So it is tough moment for all the teaching process. No? Okay. So respect lah other soft skill that we need to do more lah. Respect the patient, the parents. They are our teachers kan. Macam ni kan susah kan. Dah lah kena bawa online. Kita ada adequate opportunity to approach the patient and parents. So itulah. So next time please do it better lah. Alright.
Yeah. Doctor.
Okay.
When you say stop, I give it to her a few days and then ask her back, or stop?
Okay.
Totally.
Only lah because I cannot do for the teaching on Friday, so we just do the teaching now lah. Because the Friday I have meeting, so this week I have 6 teachings, 3 meetings, and some other admin duties lah. So basically Friday is not free. Tomorrow clinic, morning, afternoon teaching. Today actually my afternoon is free, but this afternoon you have seminar kan. Wednesday pun I have another teaching. Thursday I have morning and afternoon teaching. Friday I have meeting. So we just do it now. All right. You just present whatever you get from the parents lah. Okay. So we discuss this case. Or you prepare have other cases, tak ada kan?
Tak ada. Tapi detail saya dapat pun very simple tu. The patient I got the history from her mother, and then the patient Ilhan, 1-year-old child, boy, was come to the hospital for follow up.
Wait. Samba ni your official name siapa? Your true name tu siapa?
Muhammad Mustafa Samba.
Memang Samba ya. Okay.
Ya, nama saya.
Boleh ya panggil Samba?
Boleh.
Okay. I noticed you joined the class with group 3 kan ya last time?
Ada.
At that time discuss this case as well or not?
No, bukan this case. This lain. Pasal
yang diabetes ya?
DM ke? Down syndrome. Yang nenek pada baby tu.
Oh, yang Reski ya. This one another, yang your case ni another similar case lah, tapi another patient. More or less the same. Okay lah, just present. So we discuss.
So basically, the only thing that I can get from her reply is, the patient was come to the hospital at early June for follow up and also do the test for thyroid examination. She do the test because her son was diagnosed with congenital hypothyroidism and patent dysarthrosis since birth. Then when I try to ask further, she doesn't reply anymore. Tu je saya ada.
Okay. Alright. So what the diagnosis the mother didn't give you?
The mother just said the hypothyroidism since birth and also PDA.
Okay.
That's all.
Alright. Okay lah. So the diagnosis kan. Okay. So it's okay lah. If later the mother replied, you can try further to for your case write up lah. But for today we just discuss lah. This case is syndromic child. Yeah. What is the most common syndrome? Do you know?
Down syndrome.
Sorry?
Down syndrome.
Down bukan Draws. Ya. So Down syndrome. DOWN. Some reference just with S lah after N. Down syndrome. Okay, this is the most common one. The known case of trisomy 21. All right. So what are the features of Down syndrome?
Slanting of eyes.
Okay, Alip. One.
Yeah. We have slanting of eyes. And so we have flat nasal bridge. Also we have protruding tongue.
Okay, Samba, next.
Hypertelorism, lowered ears, and then frontal posing, lash anterior frontal, and then at the hand.
Okay.
Single farm. Okay.
So, how do you describe hypertelorism?
The increased distance between the two eyes.
Wide distance between two eyes tuh. Actually dia kenapa dia jadi wide distance? Because there is flat nasal bridge and prominent medial epicanthal fold. So medial epicanthal fold dia nampak tebal sangat ya. So jadilah dia kita sebut hypertelorism tuh. Jadi lebar sangat. The space between the interval between two eyes tuh. Okay. Nur Alia.
The hand is short, and there is a crest, a single palmar crest.
Okay, so we describe the hands. Any more from hands? Yeah, palmar crest, short, finger, apa lagi? Sometimes brachydactyly. Yeah. So the single palmar crest tu, what is the similar name dia? Simian.
Is it simian? Simian crest.
Simian crest tu, actually not only palmar, yeah, can be plantar. So that's why you must check the patient to all four limbs, ya, semua all both right and left, upper and lower limbs. Sometimes not necessarily bilateral, sometimes only unilateral, only one side only. Sometimes kat palmar ni tak ada pun, you need to check on the plantar. Often some patient ada single plantar crease. So still same lah, nama dia simian crease. Okay, Akila, can you describe more?
For the feet, there may be a sandal gap.
Okay, on the feet, spinal gap. Anything else? Yeah.
Hypotonia.
Okay, when go for the CNS, ya. CNS itu dia hypotonia. So hypotonia tu actually dia related with the hypothyroidism. Actually, the more patient with hypothyroidism, dia will develop more central hypotonia. So that's why you can see Fatin Najiha, kalau patient ada hypotonia, apa yang you can see tu? Oh, Lina. Lina tak respon pun. Dia just letak je join the meeting without participating. Tak apalah. Nuran. Yes. Mana?
Floppy baby syndrome.
So floppy infant kan. So that's why kita sebut patient itu floppy sangat. Dia will have gross motor delay, dia lambat nak head up, nak duduk pun lambat. Especially Down syndrome not only gross motor lah, all aspect of the development, all area will involve. Not only gross motor, also fine motor and as well as the speech. Okay. Other aspect of hypotonia itu, despite dia nampak floppy, lembek, kita sebut hyperlaxity, dia fleksibel sangat. So if you see the Chinese circus player tu, you nampak dia boleh pusing-pusing kaki sampai kepala pun kat belakang, hadap tengok belakang, pusing-pusing kan. Itu contoh of hyperlaxity. But not all are Down syndrome. But if Down syndrome patient is can play circus, they will play well because very flexible their extremities. Okay, another thing, hypotonia itu due to the weakness of the muscles central, so all aspect dia akan weak. So akan you nampak kenapa dia protruded tongue. Ya, itu dari hypotonia karena the patient tongue based muscles tu dia not working well, so dia tak boleh tahan lidah tu supaya tak boleh tarik lidah ke belakang, so lidah will be protruded. Ada yang sebut macroglossia kan, dia nampak besar, actually due to the hypotonia. And then another thing, due to the hypotonia of the abdominal muscles, dia lembek kan perut dia, jadi nampak distended abdomen. And then some patient have umbilical hernia. So keluarlah intraabdominal compartment to through the umbilicus. So dia sampai sini ada umbilical hernia. Itu karena abdominal muscle dia weak. So ini related hypotonia tu related with hypothyroidism. Okay, Noraisa, anything else you want to add?
For CVS, maybe congenital heart defects.
Okay, so we go for the other system ya. Tadi kemudian kita ada discuss ya. Down syndrome ni dia related with sorry. So we discuss next lah. Down syndrome ni kan is a syndrome kan. Syndrome tu means group of symptom ya. So there will be many symptoms kan. There's some external features macam tadi yang kita dah discuss. Can be also other systemic involvement. So any syndromic child, so we should roll out every systemic problem. Down syndrome ni dia other system memang banyak yang involved. Increased risk tadi hypothyroidism satu, and then CVS. Jadi apa Aisyah? Yang most common CVS or congenital heart disease in Down syndrome apa?
AVSD.
Okay, most common AVSD in theory ya. So Down syndrome very common. So I met many cases lah, not only FASD, also VSD, also ASD, also PDA. So yang this case ni siapa ni, nama Ilhan ni, dia has PDA ya. Cerita mak dia ya. Actually he has ASD as well kalau tak salah. He has ASD and PDA. So apa yang you expect, siapa Aisyah? Apa yang you expect to find with PDA? Sorry?
On physical examination.
Yes.
Murmur.
Yeah, what type of murmur?
Continuous murmur.
Okay, continuous murmur. Balik sambar lagi lah. Sambar nanti asyik tak senyapnya. Apa basic assessment kalau ada congenital heart disease, apa yang you kena assess first dulu? First assessment.
Apex beat.
Wrong lah. First color dulu, the simplest one. Color dia pink ke blue. So kalau PDA itu pink or blue? Cyanotic or cyanotic?
Cyanotic.
Ya pink lah. Patient PDA, VSD, ASD ini pink lah. Okay. So he should be pink. Then the anatomical kan, patient pink, cyanotic. Cyanotic congenital heart disease itu usually simple. Simple means only one. Kalaupun multiple pun ya simple lah, dia not related. In contrary with blue, cyanotic congenital heart disease usually complex. Kalau patient biru, dia problem dia banyak biasa. TOF itu ada 4 anomalies. Kalau still pink usually simple, only 1 or 2 lah. This patient, if I'm not mistaken, 2 lah, ASD and PDA. But still simple lah, not complex. So kalau simple nih, we need to diagnose the anatomy. Anatomi dia apa? So murmur lah who that will determine the anatomical diagnosis. Tadi saya sudah cakap kalau ada continuous murmur, kat mana Aisyah? Location. PDA.
I'm not sure juga. Is it upper left? Upper left ke?
Yes, upper left tunnel edge on the
Left tu ni.
pulmonary area, actually itu kita sebut apa? Ductal area lah, memang apa namanya? Ductus arterius area. So dia not on, memang dia actually higher than pulmonary dia. Upper left dia not only on the ICS2, but some location that we can call it subclavian. Ya. Memang betul-betul bawah apa os clavicle, ya, clavicular bone. So kat situ ya, location, and then continuous murmur ya means in both systolic and diastolic. So whenever you auscultate a patient, cardiac auscultation, before you determine the murmur, describe the murmur, first thing that you must do is determine which one S1, which one S2. If you cannot determine S1 and S2, you will never describe the murmur. So nak tahu ni systolic or diastolic, you must know which one S1, which one S2 first. Ya. So kalau systolic tu kat mana, Samba?
Macam mana? Kalau systolic tu kat mana?
Apa description of systolic murmur?
Description of systolic.
Masih senyap Samba. Poor connection tadi few times.
Dia murmur during when we feel the pulse, there's the first sound that's similar to the pulse. No.
Okay. Apa systolic murmur, Samba?
It is a murmur that happen simultaneously with the pulse.
Yang lain lah. Tukar ini pula sekarang, tukar urutan ini. Fatin tadi dah ya, belum ya? Dah. Salwa?
Yes, doctor.
Apa? Systolic murmur.
Systolic murmur is when heart is contracting. The contracting of the heart.
Itulah. You cannot see the contraction. You hear the S1 and S2 tadi. So systolic murmur will always between S1 and S2, after S1 and before S2. Itulah systolic, that's simple. So, apa beza S1 and S2 tuh? Usually S1 and S2 tuh longer or shorten than S2 to S1. Systolic, which one longer? Systolic or diastolic phase?
Diastolic.
Diastolic is longer kan. So systolic between S1 and S2 dia short. And then diastolic S2 to S1 dia longer. So recognize S1 tuh dia soft, dia suara dia lep. Kalau S2 dia louder, dep. So lep dep, lep dep. So the heart sound tuh bukan dek 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 ya, bukan one sound only, but actually two sounds. Ya. Lep dep, lep dep, lep dep. Kalau will be difficult kalau patient will have tachycardia or patient in heart failure or apa kita sebut gallop rhythm itu susah nak bezakan, nak distinguish, nak describe S1 dan S2 itu. Kalau normal heart rate senang tengok lep dep. So lep dep tu between lep and dep is systolic. After dep is diastolic. Before another lep, before another S1. So simple, systolic ya between S1 dan S2. So tak cari dulu mana nih yang lep, mana yang dep, mana yang S1, mana yang S2, baru you tahu oh this one systolic murmur, this one diastolic murmur. Ya. So kalau PDA in both lah, senang aja dengar. So, ber. So even systolic dengar, diastolic pun dengar. Ya. Oke. Okay. So still hard ya. You know it's apa SNP and then you know it is continuous murmur, so you might diagnose PDA this case ya. Patient pink. Kalau patient blue ya don't diagnose PDA ya. Even though you have continuous murmur, kalau patient cyanotic you cannot diagnose only PDA. If you sebut dia cyanotic congenital heart disease PDA, so wrong lah. PDA is always pink ya. Kalau you hear the PDA murmur, continuous murmur, but patient blue, senang aja. Actually all cyanotic congenital heart disease are complex kan. So it is complex cyanotic heart disease, CCHD. With PDA component. Memang there is PDA in the patient, but the problem is not only PDA. If patient has PDA only, patient will not blue, patient will pink. So, kalau cyanotic itu congenital ya patientnya complex cyanotic heart disease ya with PDA component is okay. Tapi if you answer that patient has cyanotic blue but PDA only, ya wrong. But sometimes case of acyanotic tuh dia kalau pink acyanotic dia not necessarily pink all of the time. Dia ada some occasion dia develop cyanotic. Kalau dia increase oxygen demand. Kalau pasien sakit terus, pasien menangis, demam tinggi atau in condition of heart failure boleh jadi cyanosis ya. Nah, kalau cyanosis dia jadi persistent, that actually congenital defect is acyanotic like PDA or VSD or ASD, but patient jadi biru terus, kita sebut jadi sindrom apa?
Eisenmenger.
Eisenmenger syndrome itu persistent pulmonary hypertension. Kalau dia patient not manage well, dia jadi heart failure, jadi pulmonary hypertension, jadilah pulmonary pressure dia tinggi, jadilah fail to carry adequate oxygen from the lung. Kalau ada pulmonary hypertension tuh dia oksigen pulmonary circulation dia tak cantik, not good, jadilah poor oxygenation nanti, jadilah dia cyanosis, kita sebut Eisenmenger syndrome ya. Tuh ya. So in CVS, any CVS diagnosis, you must consider all the three, the etiology, anatomy, and function. Yeah. So not only PDA. Yeah. So the etiology, no, it is congenital and zoonotic. Okay. So it is a PDA. If you know the patient is zoonotic, it is not PDA. It is complex zoonotic heart disease with PDA component. Yeah. Okay. And then after you know it is the etiology, you diagnose the anatomy, or it is most likely PDA. You must also assess the function. So that's why masa general examination pun, despite you tengok color dia pink or blue, tengok syndromic feature, you must also assess the failure signs, heart failure features. Kalau dia nampak tachypneic, and then need restless, cannot sleep without pillows. Kita sebut patient with heart failure. So kalau you have CVS case, you must sebut lah patient in failure or not in failure. So this case to PDA, I think they already corrected, so not in failure anymore. Okay. So it is the heart lah. Balik lagi tadi, patient despite ada hypothyroid, ada heart disease. Siapa yang belum nih tadi? Nur Alia nih dah belum?
Dah.
Syarifah yang belum ya? Syarifah,
Yes.
apa lagi other system that you might encounter in patient with Down syndrome?
GIT, because of the hypotonia. So they may have feeding difficulties also, feeding problem.
Okay, actually there are some anatomical diseases, anatomical anomalies in GI tract. Apa je? Most common apa? Atresia kat mana?
Duodenal.
Duodenal atresia is most common, but actually can be from proximal to distal. Boleh je dia develop esophageal atresia ke, pyloric stenosis ke. Another common actually di Hirschsprung ya. Most common is duodenal atresia. Another common Down syndrome tu Hirschsprung disease ya. Apa tu Hirschsprung disease? Maisara, Syarifah, or Samba?
When the intestine doesn't have the nervous system because of the due to during the development during the fetal life dia tak full the neuron tak move yet to the intestine, so the intestine has no neuron.
Kita sebut we call as aganglionic colon. So his intestine, the larger intestine, colon tu dia aganglionik, so tak cukup persarafan, jadilah dia tak boleh perform a proper peristaltic movement. So effect of aganglionic colon tu proximal from the colon yang aganglionik ni dia jadi distended, proximal, so jadilah tak boleh gerak kan, the feces tu tak boleh masuk, tak boleh pass through the aganglionic segment. So yang proximal jadi distended, all the feces collected kat sana, tak keluar-keluar lah di feces. Masa in aganglionic part tu dia usually poor, kalau kita buat x-ray dia poor air distribution, dia tak nampak sangat di udara tu. Then small part of the colon. Usually distal, can be sigmoid colon or descending colon. Jarang sangat yang ascending atau transverse. Usually descending part or sigmoid colon yang distalnya. Atresia ani pun boleh je patient Down syndrome ni. Okay. So kalau you want to diagnose duodenal atresia, apa keywords? Ya, atresia means non intact di di lumen tuh, dia buntu, dia apa namanya, blok lah, blok lumen. So
biliary,
sorry,
biliary content in vomiting.
So just imagine kan kalau dia blok, mana boleh di food, milk, anything lah, or drink or water or food tuh. Kalau in newborn kan pasti milk only or water kan. Milk tak boleh go pass through the duodenum, dia akan pergi balik, dia akan vomiting. Vomiting dia usually will greenish in color due to the biliary content tuh. So dia usually the atresia tuh distal from the biliary duct. Jadilah dia muntah dia color hijau ya. Itulah the classic finding kalau ada duodenal atresia, dia greenish in color. So but not only duodenal atresia ya, all upper intestinal obstruction ya usually have greenish vomiting, bilous vomiting. Can be jejunal atresia, duodenal atresia. So obstruction, so bilous vomiting itu sign of obstruction, intestinal obstruction. Ya. So kalau case with intestinal obstruction need urgent operation lah, laparotomy to open the obstruction tuh. So must go to the surgical, must go to the pediatric surgeon. So not medical case, itu surgical case. Beja kalau muntah sebab AGE kan, you tak payah refer to surgeon, just treat the patient with fluid ya, nanti muntah dia lama-lama hilang sendiri. Kalau obstruction, duodenal atresia, you only give fluid or any other treatment, medical treatment, will not resolve. So you must send to surgeon, must refer to surgeon. Okay. Oh ya, balik lagi tadi yang hisprang, another common ya. Hisprang tuh the common finding will be delay of meconium passage. Neonates tuh you must expect that the baby will defecate, will pass the meconium tuh within 24 hours. Usually first 12 hours pun akan dah keluar. Kalau delay more than 24 hours, we must think about some anatomical problem in the distal part of the intestine. Usually the common one is sprung. So we move on to other system. Siapa yang belum Najmuddin ya?
Yes, doctor.
Apa lagi?
Hearing impairment.
Hearing actually not really related ya, but can have lah, but not increase lah. Down syndrome not increase risk of hearing impairment. Hearing impairment tuh can be due to other usually other yang towards infection to intrauterine infection. Tapi ada je case Down syndrome dia tak ada hearing impairment. Oke, apa lagi yang lain?
Vision impairment.
Itu sama juga, actually not really related, tapi possible lah. Apalagi Samba, balik lagi lah to presenter nih.
Recurrent respiratory infection.
Okay, respiratory. Usually actually Down syndrome nih related to the immunocompromised immunodeficiency condition. Biasanya Down syndrome dia immunity dia relatively lower than normal babies, normal child. So dia lagi senang kena infection. So most common infection in children respiratory. Due to the immunocompromised condition situation. Okay, anything else? Jangan lupa tadi, yes?
Growth, development delay.
So bukan growth so development ya. Tadi kan udah discuss kalau CNS tuh neuro, usually the phrase is neurodevelopment. Kalau dia ada some neurologic problem, ada CNS situation, so dia akan developmental delay lah. Tadi kan ada discussion yang global developmental delay, all 4 areas. Ya. Cross motor pun delay, fine motor delay. Usually patient-patient with Down syndrome tuh dia due to the learning disability, dia low IQ, mental. Dulu kita sebut mental retardation, tapi sekarang kita tak sebut lah. Kita sebut learning disability. IQ dia usually less than 70 ya. Itulah dia jadi global developmental delay. Tapi growth is normal ya. So growth in Down syndrome is not affected. Unless kalau dia ada untreated tadi GI problem. GI problem kan we usually treat urgently lah. After GI tak ada problem, dia makan banyak aja. Patient Down syndrome ni other features dia is happy baby. Dia happy je. Jumpa orang tak takut. Ada makanan makan je, makan dia banyak orang patient Down syndrome ni. So growth dia usually no issue lah. Patient Down syndrome banyak je yang overweight. Kalau yang kurus jarang. So you must understand in more basic apa beza growth and development. Ya. Growth is the measured parameters of weight, height, and circumference. Kalau development kan more towards the neurologic development lah. Dia kita kena assess with milestone, with developmental assessment. Okay. Okay, anything else? Satu lagi sistem, jangan lupa di hemato-onco ya, di patient with Down syndrome actually increase risk of malignancy dia. Kita kena routinely check the MPP dia, full blood picture. Kalau ada malignant cell, ada lymphoblast or myeloblast, dia boleh jadi ALL, ML. Okay, so nanti you belajar lagi ya. Okay, anything else?
Doctor,
yes.
For Down syndrome, because the patient have hypotonia, so it is common, right, doctor, for the patient have tracheomalacia?
Yes. Another common ya. Yang tadi saya sebut actually even airway and GI tract and airway juga lah. So weakness of the muscle itu can involve in every part of the body lah. Kalau dia on the airway, upper airway dia jadi laryngomalacia, tracheomalacia tuh. Boleh. Dia more related with tadi hypotonia, related with hypothyroidism. Another thing nanti pun akan saya repeat balik, hypothyroidism tuh in Malaysia kan udah do routine screening in all neonates ya, routine neonatal screening ya. Kena cek semua TSH dia, level of the thyroid stimulating hormone. Kalau dia with hypothyroidism, dia the thyroid glands dia unable to produce thyroxine T43 itu, so TSH dia low or high?
High.
Stimulating hormone dia high, dia central hypophyse dia, hypothalamus and hypophyse dia akan increase di TSH to push the thyroid gland to produce the thyroxine. So patient with Down syndrome ni increases of hypothyroidism, dia akan banyak case-case macam Ilhan, ada Reski ya with high TSH at cord blood screening, neonatal screening dia. So that's why it is important. Kenapa? Why we need to do the screening very early at birth using the cord blood, neonatal screening. Bukan nantilah umur sebulan ke setahun ke. No, we must do neonatal screening in hypothyroidism because early diagnosis and intervention will determine the outcome. Kalau kita diagnose lambat, outcome dia lagi buruk. Ya. If we diagnose at birth tadi, the cut-off is below 2 weeks. Ya. Usually kan we send at birth, nanti keluar dalam 3 to 5 days. Nah. We dah tahu TSS tinggi, so maknanya hypothyroidism. So we must start early treatment, giving thyroxine. If we do early intervention, thyroxine will improve outcome lah. Kalau late after 2 weeks, so jadi lagi buruk. The hypotonia dia more severe, the IQ dia lower, semua jadi the developmental delay dia more severe and so on and so forth. If we do early, kita pakai betul-betul bagi cukup thyroxine. Nah, thyroxine tuh this hormone tuh is play a role in every systemic and organ metabolism. So dia pergi ke brain, jadilah brain development dia need thyroxine. So itulah kalau dia hypothyroidism, jadilah dia mental retardation. Jadilah developmental delay. Pergi ke muscle, setelah jadilah adequate muscle tone. Kalau hypotonia severe jadi macam-macam tadi kan. Laryngomalacia, gross motor delay, semua hyperlaxity, susah tak boleh jalan, banyak problem. Ya. So that's why we must diagnose early. So treatment must be started below 2 weeks. Umur 2 minggu tuh dia kena adequate thyroxine dia. Okay. Like this case pun ya, many cases of Down syndrome pun, even though we give adequate thyroxine, ya patient still have developmental delay. What more if we don't give? Different story kalau patient yang non-Down syndrome patient with hypothyroidism ya, patient has only hypothyroidism, we give early intervention, usually outcome very good. Patient can have like normal life lah, with adequate. With congenital hypothyroidism only, kita give thyroxine. So adequate thyroxine will have adequate development lah, neuro development okay, gross motor pun okay, and so on and so forth. Alright. So anything else? Okay, so sekarang lagi satu basic pathophysiology yang you must know ya. Tadi kan you must know how the feature satu, dua you must know the the related systemic disorder related to Down syndrome tadi, and explain how. And then lagi satu yang penting is the basic pathogenesis of Down syndrome. Apa Samba? Kenapa tadi saya dah sebut tadi awal-awal tadi?
Yes. Trisomy 21 dia.
Okay. The most common is trisomy 21, but not only that lah. We have 3 basic mechanism of the Down syndrome. Apa? Other 2.
Translocation and mosaicism.
Okay, yeah. So most common is trisomy 21. So when in gametogenesis lah, in gamete from the mother and father meet, and then to meiosis and mitosis proses kan. During meiosis dia fail to split ya, non disjunction during meiosis. So jadilah all the chromosome 21 jadi 3 pieces ya. Jadilah trisomy 21. So maknanya ini all the cells, all body cells, the gamete, the kalau laki-laki sperm dia, kalau perempuan ya ovum dia pun, And then all the nama dia the somatosome, all the cells of the body will be trisomy, will have 3 chromosome 21. But the mosaicism is a little bit different. Mosaicism, the disjunction tadi during the mitosis, not meiosis. So actually the gamete tuh apa namanya dia the cell of the gamete lah, the ovum and sperm tuh dia normal aja, dia akan jadi 23 kan, only one chromosome 21 kan. Tapi the body, some part of the body have 21, have 3 chromosome 21. So kita sebut mosaicism. So some part of the body normal have only 2 chromosome 21, but some part of the body have 3 chromosome 21. So jadilah actually the mosaicism tuh the feature is less prominent than yang trisomy 21. Kalau trisomy 21 jadi jauh ya first kali nampak akan tahu this one Down syndrome. Kalau mosaicism maybe not that full blown not that prominent lah. Tak nampak sangat di nasal bridge dia flat nasal bridge, tak nampak sangat the featuresnya. So usually the features is only minimal. So maybe, but we usually still recognize, oh this one like syndromic features, tapi apa nih? So we send chromosomal analysis, nampak dia mosaicism, ada yang normal kromosom, ada yang trisomy 21. Kita sebut as mosaicism of Down syndrome. Kalau yang trisomy 21 nampak lah from clinical features pun very prominent, nampak sangat ya. Another one is translocation. Oh, much more basic actually, not even before the fertilization ya, sebelum meeting of the ovum and sperm dia usually from the mother lah dia. From the mother's gamete ni dia the ovum ni dia dah the translocation. So the problem of translocation to happen on the maternal or maybe paternal lah of the germ cell ya, di ovum or sperm ni. So dia translocation dari awal. So from the maternal usually, for example ibu lah kan. So effect of the translocation kan sel ibu dah translocate, ovum ibu semua translocate. So maknanya dia all offspring, semua anak, kalau ibu ni dia pregnant, semua anak akan Down syndrome karena semua translocation. That's the difference ya. Kalau you nampak ada ibu anak dulu tak Down syndrome, sekarang Down syndrome, itu usually itu tadi yang meiosis ya, meiotic non-disjunction tadi. Usually trisomy 21 only. Kalau semua anak since first child until subsequent semua Down syndrome, ya usually translocation of germ cell of the mother. Can be father juga, but usually more common in mother kan. So it is the prosesnya. And then tadi kan usually kalau translocation adalah all, even since young age pun dia akan semua anak. But yang tadi yang trisomy 21 tadi yang meiotic non-disjunction tadi yang fail to do a junction of the meiosis process ni, usually they are related to old age ya. So kita sebut ya older age lah. Dia kalau ibu dia umur atas 35, atas more than 40 ya, usually higher risk. Or paternal juga, sometimes ayah dia umur tua, or other faktor juga, maybe radiation, maybe nutrition ya. Maybe some chronic illnesses. Ada aja patient with diabetes mellitus, anak dia Down syndrome, patient with hypertension Down syndrome. With current yes.
kalau parent kan tadi doktor kata kalau father berumur pun boleh dapat Down syndrome kan?
Yes.
Tapi kalau saya baca dia macam percentage parent kalau father yang tu dalam 5% macam tu kan?
Yeah, nah, but it's still possible kan, but less likely lah, only 5% compared to maternal apa sebut, bukan old age, kita sebut apa, maternal mature age, kita sebut dah lagi tua kan, more mature, lagi tua, actually the ovum tu dah not well regenerated lagi kan, kat mana, kat ovarium dia. Dah the one that already produced before kan, kalau dah umur more than 35, more than 40 itu actually the one existing ovum that already produced before that, bukan produce new ovum lah kalau umur dah tua. Ada some teori kat sana lah. Atau kalau produce pun itulah, kalau produce new ovum pun kalau umur dah tua itulah usually dia non-disjunction, jadilah dia bawa actually yang this germ cell from the mother, for example the ovum dia bawa chromosome 21, and then jumpa the sperm one chromosome, jadilah 3 chromosome 21.
Doktor, satu lagi. Doktor?
Yes.
Yang the one that translocation, yang bila parent have translocation tu, then Doctor said that all the children will have kan?
Yes.
Jadi dia very rare ke atau dia rare biasa-biasa? Macam doktor pernah jumpa kan beberapa kes?
The occurrence of translocation. Yes, I found a case, but not my patient lah. One case in one country lah. Dan anak semua dia Down syndrome, all the child Down syndrome, but not my patient lah. Yeah, of course the incidence itu very rare lah kan. Not all old mother will develop translocation, no lah. Only one kita sebut point mutation lah. Kenapa dia mutation sampai translocation? There are factor lah. If you learn about the mutation tu genetic kan, there are some hit factor kan, some contributing factors like radiation, termasuk actually yang common tu unhealthy nutrition. Kalau setelah beza kan different, nanti during my age pun different that during my ancient, my fathers, mothers and grandmas tu, my grandparents tu dia selalu always eat the healthy food kan. During my age pun dah starting ada some unhealthy food, what more during your age tu. So lagi ke sini more later the era tu, the more unhealthy diet tu. So this genetic issues tu will develop more lah. Lagi banyak syndrome.
Okay.
Okay. So Lina, last call.
Ya,
Ada juga. Tadi tidur, dah panggil banyak kali tadi. So do you want to say anything?
Tak ada, doktor.
What do you understand from this today's discussion? Summarize.
About pathophysiology of Down syndrome and the hypothyroidism and hyperthyroidism which should be treated early.
Heart disease apa? About the heart disease?
Heart disease related to Down syndrome ke doktor?
Okay, boleh.
The most common is ASD, but patient can also have DOM.
The most common not ASD. Siapa tadi yang jawab tadi? Yang disease tadi?
Aisyah.
Aisyah. Apa most common?
AVSD.
Can you hear, siapa Lina?
Yes.
So the most common is AVSD lah. Okay, ya. So Down syndrome tuh has apa? Multidisciplinary issues ya, multi-systemic issues ya. Even nanti approach of the management pun ya multidisciplinary lah. So we refer everywhere. According to the diseases or anomalies. So nanti you can read for now you can differences. Nanti later messenger 5 we discuss more the management. Okay. So I go back to the more specific about the CVS. Yeah. So I expect you will not get more discussion about the CVS from the other lecturers kan, because actually I'm in charge lah. So tadi I am very detailed, very basic to go for CVS. You must understand how to approach patient with heart disease lah. You must know the etiology, congenital or acquired, and then cyanotic or acyanotic, and then hear the murmur to describe the anatomy, especially for acyanotic congenital heart disease. Yeah? And then lastly for assess the function, the failure symptoms, yeah? Whether the patient in heart failure or not. Yeah? Okay. So tadi kalau macam acyanotic yang patient pink, usually simple. You must get the murmur very clear to get the diagnosis. Yeah? Actually, kalau cyanotic congenital heart disease much easier. Ya. All are complex. Ya. Actually, ya, kita sebut je, you jumpa patient blue, cyanotic, sebut. So, this one complex cyanotic heart disease. Nah. Tapi sometimes, the most common complex apa? Samba. The most common cyanotic TOF kan. So, you need to know how to diagnose TOF clinically. Ya. Ya, ada PS murmur yang you must get, pulmonary stenosis murmur, ada lifted apex, and then ada parasternal heave. Itulah you may diagnose as TOF. Kalau tak, if not very clear, not convincing to diagnose TOF, ya sebut je. Again, it is complex cyanotic disease lah. Remember for cyanotic itu remember 5 T's. Most common TOF, other common ya TGA, tricuspid atresia, apa lagi tadi, truncus arteriosus, and TEPVD. Ada 5 yang common. 5 T's tu. Actually ya banyak lah yang common, but usually nak senang ingat tu remember 5 T's. Ada juga other common cyanotic heart disease tu DORV. Apa lagi ya? Itulah. Kalau Eisenmenger is this cyanotic congenital heart disease or not? Lina aja lah, tanya Lina tadi. Dengar tak tadi? Discuss pasal Eisenmenger tadi. Tak respon lagi Lina nih. Eisenmenger syndrome tu cyanotic congenital heart disease or not? Is it cyanotic or not? Congenital heart disease.
Cyanotic.
Wrong.
So dia right to left shunt kan doktor?
Wrong. The question is Eisenmenger part of the cyanotic congenital heart disease? The answer is no. The Eisenmenger, it is not cyanotic congenital. Congenitally, Eisenmenger is come from the acyanotic. So patient Eisenmenger tu it is not congenitally cyanotic. Dia congenitally is acyanotic. Awalnya dia pink. PSD or ASD or PDA, but develop heart failure and furthermore pulmonary hypertension, jadilah dia Eisenmenger cyanosis. Indeed, the feature is cyanotic. The patient is blue, but it is not cyanotic congenital heart disease. It is come from acyanotic congenital heart disease that not well managed, develop pulmonary hypertension, and later develop Eisenmenger. Okay. Yeah.
Okay, Doktor.
So you must understand the pathophysiology of the congenital heart disease. Ya. Nanti every single detail about features of the murmur you belajar sendiri ya. We don't have adequate time. We need to stop at 12 because I have a meeting at 12. Okay, the features tadi yang apa? Features of apa namanya? Group list of the disease of cyanotic heart disease apa aja tadi you belajar satu-satu. Yang cyanotic congenital heart disease apa aja tadi you belajar satu-satu, the findings, the murmur and so on and so forth ya. And then if it is not congenital kita sebut acquired kan. Acquired itu means congenitally ya dia tak ada sakit jantung tapi didapat jantung tu sakit. Most common acquired apa? Tambah.
Allah, ya.
Yang lain?
Sure.
Others?
Ineffective endocarditis, Doktor?
IE is not most common. Apa? Most common acquired? IE itu infection lah. Biasanya namanya very poor hygiene, not treated well. Tak pernah datang dokter barulah jadi kena IE dia.
Rheumatic heart disease.
Most common is rheumatic heart disease, secondary from rheumatic fever. Jadi itu related to poor economic status lah. Ke apa rural area, kayak Kelantan masih banyak. Kuantan pun bukan Pahang pun not in Kuantan, usually from Muazzam, from Rompin. Dia memang poor hygiene lah, dia poor economic status dia. Develop rheumatic fever. Usually rheumatic fever kan from tonsillitis not well treated, streptococcal tonsillitis dia kena streptococcal infection, jadilah dia develop rheumatic fever. Rheumatic fever if still well treated jadilah dia rheumatic heart disease, some sequel usually common ya, mitral regurg and aortic regurg. Nah, itu most common. So you must know that rheumatic ada ini kan, ada present kan dulu kan. Present dengan saya kan ya? Seminar. Tak ingat dah.
Dr. Ismail, congenital.
Oh, dengan itu. Bukan rheumatic.
Itu congenital kan.
Rheumatic fever with me lah. So the one rheumatic heart disease, rheumatic fever ni dia you must know it is the age usually more than 5 years. The peak incidence is 5 to 12 year old. So kalau you jumpa patient 1 or 2 or 3 years old, jangan diagnose rheumatic heart disease ya, don't diagnose rheumatic fever. You diagnose rheumatic fever, rheumatic heart disease itu kalau patient 5 tahun atau lebih, usually school age lah, dia ada budak sekolah. Besar ya. Because streptococcal infection itu yang more related to itulah poor environment, poor hygiene, dia lagi besar budak. Kalau baby kan masih duduk kat rumah aja. Okay. Tapi ada some acquired heart disease in younger age. Kalau umur setahun, sometimes infant pun dia ada some acquired heart disease, lah. Ada dia sakit di itu dia underlying causes tadi no with poor hygiene jadilah di IE, baru jadi IE. Okay, alright. So itu ya, so that's it. Actually the heart disease itu only that limited ya, and then very rarely has some update lah. This knowledge of heart disease itu even since 10-15 years ago when I study medical entering medical program like you kan sama aja, tak berubah banyak pun. The diagnosis still the same, how to diagnose still the same, how to manage still the same. Maybe just a little bit difference in the incidence. Kalau dulu masih banyak rural area, banyak case rheumatic fever, sekarang dah more upper middle income tuh rheumatic fever udah kurang kan, reducing. Kalau macam itulah, and then increasing number, dulu Kawasaki disease tak banyak orang tahu, sekarang udah banyak orang tahu. Kalau macam in urban area in KL tuh udah tak ada dah rheumatic fever, rheumatic heart disease. Yang ada from Rontin, from Muazzam, from Kelantan, from Musang, orang asli. But the diagnosis still the same, the management still the same. Okay, so tak banyak sangat beza. So you learn once pun you will understand and you can easily understand and you can use it all the time. Unlike kalau macam immunologic diseases yang ada immunologic itu sometimes ya very high frequency of update. Treatment dulu dengan sekarang pasti lain-lain. Okay, so itu you belajar heart tuh, and then another yang final message from me to learn about the heart disease tuh, please don't memorize. Don't only use kalau VSD pasti systolic murmur on the left sternal edge. So you must understand the key word to understand heart disease is understand the hemodynamic, the blood flows, the pressure of the heart, the hemodynamic, how the route of the blood flow, systemic circulation, pulmonary circulation, to left to right sun, when to develop right to left sun. So only then you can understand the heart disease very easy. Kalau acyanotic, you must be left to right sun. So dia pink karena left heart pressure, left ventricular pressure higher than right. Tapi kalau yang cyanotic, kenapa dia blue? Karena right to left sun. So bukan karena congenitally dia right ventricular pressure higher. Ada some condition that forced to make higher right ventricular pressure. Contoh macam TOF kan, due to the pulmonary stenosis, dia difficult to go to pulmonary circulation, jadilah dia right ventricular hypertrophy. Karena right ventricular hypertrophy jadilah higher pressure on the RV than LV. Jadilah dia right to left sun with VSD dia. Belajar tuh ya, so kenapa you dengar murmur tuh systolic, ya due to the contraction kan of ventricles. Kenapa diastolic? Due to regurgitation usually. Kalau aortic tuh ya regurgitation jadi diastolic. Kalau mitral regurgitation ya still systolic. And then location ya determine where is the defect. Kalau VSD ya lower left, kalau ASD upper left, pulmonary stenosis upper left, tadi PDA pun upper left or subclavian. Kalau mitral regurg on the apex. So as long as you know the hemodynamic, you learn, you will know lah the story. Itulah my take home message. And then kalau you not interested sangat to understand tuh, you baca dulu yang ASD dulu. Kenapa ASD ada fixed split? Kenapa ada ejection systolic murmur on the upper left sternal edge itu? You must know the reason. Nah, itu baru you paham the reason of ASD itu, yang lain senang, nak belajar. Okay, so open pitch protocol is very simple. Kalau for heart disease, better you open the textbook lah. Not only Nelson pun still general kan, all the aspect of the pediatrics. Better you go to pediatric cardiology textbook. Ada aja kat library itu. Many textbook itu banyak. Tapi kalau nak baca the simplest one, yang easy to understand the language, how to explain tuh. Baca kan buku, kalau buku tebal lagi tebal macam Nelson lagi pening nak paham kan. So yang simple tuh kalau pitch cardio tuh belajar yang PAK tuh. The PAK tuh the author lah, Myung K Park ya namanya. Korean doctors who live in America. Baca kardiologi itu can understand tadi the pathophysiology, kenapa ada murmur ini, kenapa ada defect itu. Kalau baca yang lain pun boleh. Ya. Okay, so I think that. So please keep updating your knowledge, because what more in this online session, you have very limited opportunity and to practice your skill kan. Kita ada masa untuk skill, masa interviewing long case clerking skill pun susah juga, ada yang susah. Tadi ada mak yang tanya video call ke tanah direct call, so very limited. So your knowledge ini kena baca banyak-banyak kan. You udah have more than 3 months MCO, kenalah baca banyak-banyak, belajar banyak-banyak. 5 months MCO just main game je, tidur je, tengok YouTube je. So you don't have any update, ya susah. Macam yang hari itu kan ada yang dah lah MCO 3 bulan, nak present seminar Dr. Ismail pun minta postpone pula. For me it is ridiculous because you have 3 months to prepare your seminar presentation. So that's why kalau sekarang ada macam Dr. Alwi tadi ubah nak awalkan seminar macam tadi pagi, ya you must be ready at any time, because you have very ample time to prepare your knowledge, prepare your seminar, your knowledge buat the diseases. Bacalah banyak-banyak ya. So you dah tak ada masa untuk praktis, tak ada masa untuk jumpa pasien, you kena baca banyak-banyak. You kena compensate lah. Kalau dah jumpa pasien tak dapat, baca pun malas. Susah lah nak upgrade your knowledge ni. It is not my responsibility to make you understand. It is not our lecturer or even university responsibility. Our responsibility is to assess lah. If you don't qualify, you just fail and then repeat lah. Okay, so please update your knowledge as much as you can, supaya you can catch up your gap nih, because you have very huge gap compared to other medical students yang dulu yang ada, terus even yang from your previous blogs pun lain kan. Medical dulu masih sempat belajar sebelum after medical surgery ya. Sebelum medical you buat surgery ya. Surgery medical udah sempat belajar, tapi pediatri tak sempat. Nanti ONG pun you will have similar condition situation. Tapi you pray juga lah. Most likely next month you can return to Mahalah, to go back to Kuantan, because yang even high school primary school pun dah masuk pula semua. Takkanlah yang GPT asyik kat rumah je, duduk kat rumah je. Most likely next month you will be called. Okay, catch up ya. You have very huge gap lah. So belajar banyak-banyak, tengok baca buku banyak-banyak. Maybe another suggestion, tengok video banyak-banyak, tengok YouTube untuk tahu apa-apa tadi. Tadi basic murmur pun tak tahu. Even the systolic and diastolic pun tak tahu, S1, S2 pun maybe tak tahu. Tengoklah banyak-banyak di video di YouTube. Ada some application juga untuk heart sound ada. So push yourself. Then itulah the only way to push yourself. Maybe you need to discipline lah. From 8 to 5, you please don't do any other activity than study because it is your time kan. Kalau kat sini kan 8 to 5 you kena ada kat hospital belajar atau kat campus kan. kat rumah you push. And then you have more time kan kalau you Selasa belajar tengah malam you can learn. So don't waste your time lah. Alright. Anything else? Already 5 minutes past 12. So we close with tasbih kifarah and asar. So, okay. So please forgive me for any mistake. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam.
Thank you, Doctor.
Thank you, Doctor.